Let's go ahead and get started. So today is Tuesday, February 2nd, Groundhog Day. We are going to finish off the last little bit of lecture we have. Again, we've tried to front load this as much as possible with a Thursday exam, but we do still have a couple major physiological processes to talk about today. And being the smart, sophisticated students that you guys are, knowing that protein synthesis involves basically two related processes, how many possible essay questions does that at least lend itself to?
Three.
Three, excellent. So I would say three. What are the three?
Compare and contrast.
There you go, exactly. Describe process one, describe process two, and then some type of either comparison or contrast between the two. How are they similar? How are they different? Things along those lines. Absolutely. So those are the types of questions you should be asking yourself as we are going through today's material. We won't go the whole lecture time today, so that should leave us plenty of time for a review for the exam on Thursday. Again, a review is not me standing up here telling you what I think is important. That's what I do every day in class. A review is for you to ask questions of me, and we will work out the answers together. And again, if you guys don't ask questions, then I assume you've all mastered the material and I make the exams harder. And the exams are on Thursday. It's our first exam. You've taken multiple choice questions before, you've answered essay questions before, and thanks to last week, you have seen what my lab exam is going to be like. So there shouldn't be any surprises on this. You know what to expect. Both exams are timed. We'll have a specific amount of time you need to complete them within, and they must be completed during class time. They won't become available until noon, and at 4:35 they shut down. So even if the lecture exam has 2.5 hours on it, if you started at 4, you're only getting 35 minutes until it shuts down. So make sure you complete it during class time. Just like if we were in the classroom. If we were in the classroom and the class ended at 4:35, you couldn't come strolling in at 4 and start your exam at that point because another class is going to come in shortly after us. So it must be completed during this class time. Again, you can take the exams in any order you want. If you're really worried about the essay questions, it's okay to take that one first. You can take them in any order. But while the problems we've had with these have been limited, and they've definitely gotten better over the past year that we've been doing this, most of the times when I have a student that has a problem, it's usually during the lab exam, just because the sheer volume of material with, again, this exam, I think somewhere between 60 and 70 questions, somewhere on there. I haven't finalized it yet. Just downloading all those pictures at once can be taxing for some computers and some interwebs. So if you have problems, it's usually on the lab exam. So I recommend you start with that one, but it's not required. Then you have a whole 5 days off, and during that time, I want you thinking about the new material. As I mentioned and warned you about already, this next section is a class that is a section that typically is 3.5 days worth of lectures. And because of the ways the holidays fell out, we are only getting 3 days for it. So it is going to be fast paced. It is the least intuitive amount of material that you're going to cover. We're going to be doing all the tissues, and even the skin is a lot of microscope work. And again, not something that's as intuitive. And so to encourage you to be thinking about that material, the least you are going to be responsible for is doing the 2 pre-labs. Again, pre-labs, not the reviews. In your lab manual, there are basically 3 parts to it. There is the reviews at the end that you've been turning in for each unit. In the middle are all of the activities that you can be doing. And at the very beginning of each unit is a pre-lab. It's typically vocabulary, labeling activities. You should be doing those anyway because they're great study tools to help you to master the material. But in this case, you are going to be turning those in. And like all assignments, these are due at the beginning of class. All right. Questions on any of that?
Question. So you said
we only get 3 meeting times before next exam. Is that correct, or did I misunderstand you?
Yes, our exam is Thursday the 4th, and your next exam is 2 weeks later on Thursday the 18th.
Okay,
So
and
that means we have 3 lectures to cover that material.
I have a quick question. Do we want to read ahead to the chapters and the materials we'll be covering? Are they chapter 4 and 5?
Absolutely.
Are they 4 and 5?
It's cell, I mean, it's tissues in the integumentary system. So whatever that is. Yeah, 5 and 4. No, whoops. Where's 4? There's 4. Yeah, tissues and the integumentary system. So it looks like that's 4 and 5. Yes.
Okay, thank you.
Yeah, I tend to not put the chapters that we're doing on the syllabus and things along those lines, because every time they come out with a new edition of the book, they sometimes juggle those things around. And so to avoid confusion, I focus more on the topics we're going to be covering than the chapters, but it does look like those are the chapters for that. Yep.
Okay, so it'll just be on those 3 days of lecture, right? It won't be on the stuff that we've been doing already? Or will that be in there too as well?
Well, so again, what you got to remember is we're building the foundation of things. One of the things that we're going to do when we throw an epithelial tissue and a connective tissue together is we're going to make a membrane. And haven't we already talked about at least one membrane in this class?
Yes.
Yeah, there's actually going to be 4 membranes we're going to actually talk about in that next section. So, and of course, tissues are made up of cells and things along those lines. So yes, these first couple sections, these first 2 sections really are the building blocks, not just for the rest of 430, but for all of 431 as well. So, and again, that's one of the reasons we have a cumulative final exam. Once you take that exam on Thursday, you can't just forget all that material. Okay.
All right.
Yeah. So for instance, if I ask you, like, what glands would you find in, for what skin glands would you find in the brachial region of the, body? Is that a fair essay question on the exam? Absolutely, because you need to know what the brachial region of the body is, or compare the difference in the glands between the plantar region and the brachial region. Is that a fair question to ask on the exam? Absolutely, because you need to know those regional terms.
Okay.
Yeah.
So just make sure you study everything.
Yeah, just know everything. That's all. All you have to do to be successful in this class is know everything. It's not that hard. Very simple, right? Yes. Did you have another question, Yulia?
Yeah, I have another question. So for our labs, do we have more information we can get to maximize them? Like I don't think we've had anything assigned from our histology or photographic atlas, but would there be helpful material there for preparing for this exam?
This one, no. Well, not necessarily. Okay. I know your histology will have mitosis. So remember we talked about mitosis and that process. So are there going to be some good histology slides in there for that? Sure, absolutely. However, when we get to the tissues and we get to the skin, is that histology atlas going to become a whole lot more important in the next part of the test? Yeah, absolutely, in the next part of the class. So yeah, I mean, it's not useless right now because there is at least the mitosis stuff in there, and you definitely could be responsible for images like that, but we haven't built up really to the microscope work yet. That's what we'll be doing in the second part of the class.
Okay.
Yeah.
And then I have one not related to the exam, but just out of curiosity, we talked briefly about mitochondria RNA having, we inherit from our moms.
Mhm.
Does that have any direct effect on our abilities or our genetics? Kind of like, would it affect our metabolism, or is all of that taken from both of our parents, and the mitochondria DNA only accounts for like cell functions, not physical?
The mitochondria DNA is solely functional for the mitochondria.
Okay.
Okay, so just for making mitochondria, not for anything else. So if there is a mitochondrial disorder that your family has, there are some mitochondrial disorders where people don't make enough mitochondria, or they have abnormal mitochondria, or some other types of problems with their mitochondria that way. That would be something that would be inherited maternally. So it wouldn't be something that you could get from your father. It would only be something you would be able to get from your mother. Yeah. All right. But yeah, that would be the only thing it affects. It just affects the mitochondria. It doesn't affect anything else. Great questions. Any others? All righty. In that case, onward and upward. So, as I mentioned, our last physiological process we need to talk about is protein synthesis. Right. After all, we talked about how vitally important proteins are for the body, for the cell, I guess I should say, and so therefore for the body. So making those proteins is something that is going to be vitally, vitally important. Now, in biology, there is this rule that one gene produces one protein. How many genes do you have in your DNA? Does anybody know? We used to think we had close to 1 million genes. Does anybody know closer number?
10,000?
A little bit more than that. So, but you're right. It's in the tens of thousands, around 20,000 plus genes that we have that are responsible for making primarily proteins in our body. However, as we learned when we talked about the structural organization of proteins, right? The sequence of amino acids, those the beta sequences, the alpha sequences, the secondary, which gives the tertiary structure. And remember, most functional proteins are made up of 2 or more subunits. So remember, we saw how collagen is made up of 3 linear subunits. Hemoglobin is made up of 4 subunits, 2 A subunits and 2 B subunits that come together to make our hemoglobin. So it's actually more accurate to say that one gene gives us one sequence of amino acids, one polypeptide. And then we take multiples of those polypeptides and put them together into functional proteins. But that's really the key. One gene, one sequence of our genetic information. That gene gives us the information for making one sequence of amino acids. Now, again, a polypeptide, we're talking about a sequence of amino acids that are thousands of amino acids long. So, I mean, these are big, long strings of amino acids. But that's what a polypeptide is. And as I already hinted at, there are 2 steps involved in this process. The first of these is transcription, right? We have this issue. This issue, if you think about it with our cell, let's go ahead and quickly draw a cell on here. Where is the DNA?
In the center.
In the center? Well, okay, can we be?
I mean,
I'm sorry?
would it be in the nucleus?
Yeah, absolutely. Yeah. It's going to be inside the nucleus. Now the nucleus can be in the center. You got the right idea.
It's early. Okay.
The DNA is going to be found here in the nucleus. And all of the equipment to make proteins, and what's the primary structure that is responsible for making proteins?
Would it be ribosomes?
It would be ribosomes. And where are most of those ribosomes found?
Well, they're kind of everywhere, but wouldn't it be the rough endoplasmic reticulum?
Yeah, there are some free floating, you're absolutely correct, but most are found bound to the rough endoplasmic reticulum. And again, I'm going to abbreviate it because of space and time for that. Most bound to the rough endoplasmic reticulum, which is out here in the cytoplasm. And so that's an issue, right? That DNA, when we need that information to be able to make a protein, right? Does the cell check it out like a library book by giving that information and letting it be taken out of the DNA to go to the ribosomes, and then it gets to be brought back a couple weeks later? with a 10
No.
cent fine if it's a day late or anything like that? No, in many ways, this is very similar to in medieval times. Medieval times, right, books were valuable. They had tremendous amount of important information, and knowledge was power. So you couldn't go up to the nearby castle or a nearby church with your library card and check out a book, right? First, you had to be deemed to be worthy of that information. And even if you were deemed worthy of that information, would they then give you the whole book to take with you? No, they would have individuals who would make a copy of just the information you wanted, a temporary portable copy. Of that information, and they would give it to you. And who were the people who made those temporary portable copies? What were they called? Scribes. There you go. Madison's got it. Right? And that's what transcription is. They were scribes. In fact, one of the interesting things about many medieval scribes is that they were illiterate. They couldn't actually read. What would happen is they were given a book to make the copy of, and they would see whatever was in that book, and they would make a copy of it letter by letter. But they had no idea what it meant, because if your scribes could read, then that information could get out, right? And you would lose your power, you would lose your strength. So many scribes were illiterate. So basically, transcription is this process where we make a temporary portable copy of the information. And notice also, it is still in the same language. And in this case, that same language is nucleic acids. If you could go back in time and you could go to a castle or a church and ask them for a piece of information, and they deemed you worthy and they made you a copy and gave it to you, would it likely do you any good right now? What language was it most likely in? If you think of medieval times, knowledge was in what language?
Latin?
Latin, in Latin, absolutely. And how many people here read Latin? Anybody? I know we all know carpe diem, but other than that, anybody know any other Latin?
I just
No, exactly. Oh, Cody's knows some. Excellent. Perfect. So Cody can translate it all for us. But for the rest of us, we need to convert that language. We need to convert it from the language of DNA, nucleic acids, to the language of proteins. And what is the language of proteins? What are proteins made up of?
Amino acids.
Amino acids, exactly. And so that is step 2 in the process. Translation, where we change the language. We're going to change the language from nucleic acids to amino acids, which in and of itself is tricky. We'll talk about this, but let's talk about things we already know. How many total different letters are there in the nucleic acid alphabet? How many different nucleic acids are there? No, 5. There you go. You're right with the 5. 5, absolutely. Give me the letters.
AT,
AT,
CG,
CG, and
You.
U. Perfect. Excellent. Right. Excellent. Anybody remember how many total amino acids there are?
20.
Yeah, 20. So notice A can't stand for amino acid 1 and C can't stand for amino acid 2. We're going to have some issue here. Translation is going to be a little bit of a tricky problem, but it's one we can absolutely solve. All right. And of course, this is going to occur in the cytoplasm. So I've got to erase my picture here so we can get a prettier picture like this one here. That basically talks about these 2 processes, transcription and translation. Now, we are going to do transcription first, so let's talk a little bit about that. And to do that, we need to talk a little bit more about our nucleic acids. We do know that there are two main types of nucleic acids, DNA and RNA. And we also know that there are 5 total nucleic acids, but does DNA use all 5? No, it only uses 4. Which 4?
AGCT.
Exactly, AGCT. RNA also only uses 4. And what are the 4 it uses?
It uses uracil, but I
Ins-
forgot instead of what. And the other 3.
You're right, it uses uracil, but what does it replace with uracil? Anybody remember?
T.
T, the thymine, exactly. So it also uses A, it also uses G, it also uses C, but remember it uses uracil instead of thymine. Here, and again, we are going to then make a we can then, the advantage of this, let's say it this way, is we can still use our complementary base pair rules. And what were our complementary base pair rules again? So A went with
A and T.
A
C.
goes with T, C goes with G, right? And so then for RNA, it's going to be A goes with U, and C still goes with G. So we're still going to use those same complementary base pair rules. And remember, during DNA replication, we had a special enzyme that could use those DNA pairs to make a complementary strand of DNA. And it was called DNA polymerase. Well, guess what? We have an enzyme that can use complementary base pair rules to make a strand of RNA. And we call it RNA polymerase. See how that convenient that is? DNA polymerase makes a strand of DNA. RNA polymerase makes a strand of RNA. So this enzyme, again, along with dozens of others, but this is the primary one that is responsible for making that complementary strand. And again, here's how it works. Remember, these are our base pairs. One purine goes with one pyrimidine, adenine goes with thymine, guanine goes with uracil. And if you take a look, you can see that uracil, Ariana, you just have to memorize it. You have to memorize these base pair rules. We've talked about it a couple times. If we go back, remember, we understand the reason why. There are basically 2 factors that determine the base pair rules. We always have one large, right? The adenine and the guanine, remember, are the purines. They are a nitrogen base is made of 2 nitrogen rings. And they always go with a single nitrogen ring, a nitrogen base known as pyrimidines. But what determines which one goes with which is because the adenine and thymine are both capable of producing 2 hydrogen bonds, whereas guanine and cytosine are both capable of producing 3 hydrogen bonds. And so again, this is just one of those rules you have to remember. The way I do it, my very first girlfriend, her name was Candy Gagne. So her initials were CG, so I know C and G go together. So if you also dated her, then you may also use that as a way to remember this. All right. Excellent. Now, notice as we talked about, see, uracil looks exactly like thymine, and or very similar, not identical, but very, very similar. And so not surprisingly, it's the one that matches up with adenine when we are making RNA. All right. Now, this is a 3-step process. And like many processes, I want to go through it first on the whiteboard so we have a chance to talk about this. And again, for this, I encourage you to just sit and watch and think and listen. So what we need is a strand of DNA. This strand of DNA is what we are going to call our template strand. This is the one, and that's way too big. 18. This is our template strand. The template strand is the one that contains the information for the gene. And so for this, we can make a strand of DNA. No, no. I want that 18. I want this 36. Capital. All right. Excellent. Yeah, I'm all for mnemonics. Anything that helps people to be successful is something that is definitely useful and worthwhile. And I appreciate people who share that. Excellent. All right. Now, one of the things we know about DNA is it doesn't just hang out by itself. There is always going to be a complementary strand. That complementary strand is what, oops, don't want that to be all capitals. A complementary strand is also what is known as the coding strand. So you may hear it referred to both ways. And so, of course, using what we know to be the complementary base pair rules, we can then figure out what would be complementary here. So what would be, gosh darn, undo, let's do that again. Excellent. So using our complementary base pair rules, start walking me through it. What would be here?
G.
Yep.
E.
Yep.
A.
Yep.
T.
Yep.
G.
Mm-hmm.
C.
See, there you go.
T.
G. Yeah, and then I can go through the rest. You guys don't have to keep
Okay.
it, but I'll get the rest of it. TA. Oops, that's not gonna work. And actually, you know what? I'm going to cheat and just go ... Perfect. All right. Excellent. So that is our complementary strand, our coding strand. Now, we have an issue here. The issue is we are a cell and we want to make a protein. And as we talked about, proteins have over 20,000 genes in them. Pretend that's 1,000 genes in them. However, those genes only make up about 1.5% of the total DNA. We have a tremendous amount of what used to be referred to as junk DNA. However, it is a very new field of study over the past 10 years or so, where they're looking at epigenetics, what some of the overriding effects of all of this, all of these junk DNA and repetitive patterns of DNA may be. And it's a tremendous, interesting field of study. I actually have some friends from graduate school who are doing work on that, but none of it helps us with protein synthesis. So we won't worry about that. What we do have an issue, though, of is if I were to throw a dart at my DNA, what are the chances that I'm going to hit any protein, let alone the protein that I want?
Small.
Very small, right? So we need some help. We know we have this special enzyme, and I'll just draw it as a block here. This big globular protein. And this big globular protein, as we mentioned, is our RNA polymerase. And so we don't just want it starting at any old point. So the first step in transcription is a process we call initiation. And that step of initiation is where we have to find a special sequence of DNA known as a promoter region. There are several promoter regions, and I'm not going to make you memorize them, but they are often things like AT, AT, AT. So notice as this RNA polymerase is moving around, as it's looking at our template strand, lo and behold, it finds that promoter region, AT, AT, AT. And at that point, it is going to separate the two strands, and it is going to bind to this DNA. Now, once it binds to the DNA, then step 2 is going to be elongation. Elongation is where our RNA polymerase moves down the template strand making that temporary copy of the DNA. But it does this not with DNA, but it uses RNA nucleotides. Of course, this means that it has ribose as the sugar. And any place you would normally put a T, it puts a U, a uracil. So as this is moving down, and I'll cheat and move it to this side, as this is moving down using RNA polymerases, so for this we'll change the color to blue, what would our complementary RNA base be here at this first spot?
GCAT.
Okay, well, then slow down. So G, I heard C, I heard A. What would be next? U, right? And just remember, instead of using T's, it uses U. What's next?
G.
Yep.
C.
C, yep.
A.
A.
U. U. G. G.
Mm-hmm.
C.
Oh, what the heck's an E doing in there? Yikes. All right. let's pretend it was a C. So we'll put a G for there, and so on and so forth. You got the idea down the list, okay? Now, is it going to continue to do this just till it gets to the end of the DNA? After all, someone remind me, how many strands of DNA do we have?
2.
I have more than 2. Our DNA, remember, is a double helix. but it does have more than 2 strands. How many strands, individual strands of DNA are in a nucleus?
Oh, it's the number of chromosomes.
Well, exactly, because with each one is going to fold up to make a chromosome. So how many chromosomes would you have?
46.
46, exactly. So there's 46. So notice you've got 46 what's going to be chromosomes, but right now is just loose chromatin. And notice only 20,000 genes, or over 20,000 genes. So each chromosome has thousands of genes on it. So I keep saying 20, I mean 200,000, I'm sorry. No, that didn't sound right. Each gene has thousands of proteins, genes on it. Each strand of DNA has thousands of proteins on it. Thousands of genes. So once it locks on, do we want it to go all the way down to the end?
No, you want it to stop somewhere.
No, you want it to stop. So the third step in this process is termination. And that termination is where we hit a termination sequence. That termination sequence is often something like AAATTT or something along those lines. At that point, what ends up happening is our RNA polymerase lets go of the DNA. And one of the keys to this process is the DNA is unchanged by the process. Unlike when we replicated the DNA, we separated the 2 strands and we made new copies to go with them. As the RNA polymerase moves down, as the RNA polymerase moves down the line, our DNA comes back together. So at the end of the process, our DNA comes back together is unchanged. Our RNA polymerase goes away. And we have this sequence, a single sequence of RNA nucleic acids, nucleotides, left. Once this is left, it needs to be modified. It needs to be cut and shortened. Pieces need to be taken out of the middle. So at the end of this, we may end up with something like, let's go CAUGCA GGC, something like that. So it needs to be modified. And once it's modified, then we finally have our message that is able to leave the nucleus. And of course, we know when it leaves the nucleus, what sits right next to the nucleus?
Endoplasmic reticulum.
Yeah, the rough endoplasmic reticulum. So it's going to leave the nucleus and go to the rough endoplasmic reticulum. And this single strand of RNA nucleotides that can now leave the nucleus and go to the rough endoplasmic reticulum, we call messenger RNA, which after you've written out messenger RNA once, you can abbreviate it mRNA. So I guess there's kind of a fourth step, and that fourth step is modification of the messenger RNA.
I have a quick question.
Yep.
Why does the RNA nucleotide need to be modified? I don't remember I don't think I caught the explanation.
I didn't explain.
Oh, okay.
I don't have a good reason as to why.
But it needs to be modified before it can make a protein.
Yes, before it becomes functional, it needs to be modified. Why is always one of those tricky questions in anatomy and physiology, right? The engineer that designed this whole thing didn't leave us her footnotes, but we can often infer why things occur the way they do, but I don't have an answer for you for this one. My guess is maybe it has to do to reduce the amount of abnormal messenger RNA that gets sent to the ribosomes, so we don't make abnormal proteins, but I don't actually have a reason. I don't know why.
Okay, thank you though.
Yep. No, it's a great question. I don't have a satisfactory answer for you, but it's an excellent question to ask. Absolutely. All right, we did it on the board. Let's look at this with the pretty picture and do the same thing. Again, we have our special enzyme, the RNA polymerase, that is going to, and again, keyword here, temporarily unwinds and copies the DNA. So the DNA is unchanged by this process. And it requires 3 steps. Step 1, as we mentioned, is initiation, where our RNA polymerase finds that promoter region, a special sequence of nucleic acids that tells it this is the beginning of the gene. Notice here we have our 2 strands of DNA. One is the template strand, which again has the information we want, whereas the coding strand, remember, is just the complementary strand of it. It may have information for a different gene, but it's not the gene we want. So here we bind on our promoter region. That is step 1. Once it binds on, then just like you learned in the last class, just like we did at the beginning of this class, using our complementary base pair rules, we are going to make a copy of our template strand using RNA. Now, this is a really simplistic illustration. I do really like the picture in your textbook of this process of elongation, right? Our RNA nucleotides are being added. Notice as it goes, the DNA comes back together. It coils back together. It is unchanged by this process. And notice also right here, I'll go ahead and draw a circle around it. In this region is where we're doing all of the work. We're using the complementary base pair rules to add C's to G's and A's to T's and U's to A's and so on and so forth using those rules. This region where the work is being done is what we call the RNA DNA or DNA RNA hybrid. So again, this is a simple illustration. Let's look at the pretty picture from your textbook. Here's the pretty picture from your textbook. Here is that globular RNA polymerase. Notice here is where we're unwinding the DNA. Here is where we're putting the DNA back together. And here, using our complementary base pair rules, we are adding RNA that matches the DNA. And notice the way that they've emphasized this for us. Notice they've used purple to represent the deoxyribose sugar backbone of our DNA, whereas they have used blue to represent the ribose sugar backbone of our RNA. Oh, hold on, looks like there's a question. Excellent, absolutely. This is not off topic at all. I have no problem talking about COVID. You are absolutely correct. Well, this part, no. This is not the part that we're doing, but you've got the right idea. Protein synthesis is what the new COVID vaccines are about. They are about making an RNA strand, just like we're making an RNA strand here, that we can send to the ribosomes so that the ribosomes can make a protein. Exactly. So the same way we are making a protein that our cell needs, right? If this is a toenail cell, then we're making keratin to make the toenail hard. And so we need that material. Well, what they've done with the COVID vaccine is they've made a strand of RNA that makes one of the proteins on the surface of the coronavirus. As we talked about when we talked about the plasma membrane, we recognize things by the proteins on their surface. So the goal of the vaccine is to get the cells to make the proteins. So when that protein gets released out of the cell, our defense cells can find that protein, recognize the protein, and build up a memory to that protein. So that if you come in contact with the real COVID virus, right, you're not being infected with the virus, you're just being affected with the protein. Well, you're not even being affected. Your body's being tricked into making your own defense. So your own memory, so that you recognize that, so that if you ever come in contact, you've got memory cells and can produce a much more vigorous immune response to that. So yes, so the transcription is not a part, but translation, which we're going to talk about next, is exactly what those messenger RNA COVID vaccines are doing. Yes, Yulia.
I have a question. So when people have genetic disorders or autoimmune diseases that seem to set in after birth, so it didn't happen during fetal development, is this by any chance like a problem that occurred during DNA duplication or RNA, or no?
It's a great question. Part of the problem with immune disorders is we don't understand or know the causes of them, right? Sung, I'll answer your question in a second. Most autoimmune disorders, we don't understand the causes. One of the things that we have learned is there does appear to be a genetic predisposition. What we found is that it typically takes multiple events to occur in close proximities. There may be environmental factors, there may be viral factors, there could be other things that are involved in these things. And if they occur together, that can cause one of these issues where the body suddenly starts attacking itself instead of, or responding to a not dangerous protein that it wouldn't normally respond to. Why that occurs is not fully understood. We do know there appears to be a genetic component to it, but it isn't so much that you have a mutation to your genes that you can pass on to your kids. It is more that you have a gene that predisposes you to having that. So there may be a genetic component that makes it more likely that if those events occur, that it may happen. So some autoimmune disorders are slightly more common within families because of that predisposition. But most the causes, I mean, that's part of the problem. We don't know what causes. If we understood better what causes many autoimmune disorders, we'd be much better able to attack it and defend it.
Okay, but it's not traced back at all to the DNA code unwinding and rewinding and
No, not from anything like this. No.
Okay, cool. Thank you.
Yeah, well, predisposition for heart disease, all sorts of, there are definitely diseases and disorders that have a genetic component to them. Something as simple as color blindness to cystic fibrosis. Others have predispositions to them, like rheumatoid arthritis or things along those lines. Sung, so again, we will talk about this in much more in 430 and get into much more detail, but here's the short version. Okay, let's start with this. The way we respond to getting sick is our body builds up a defense to that sickness. It takes time, about 10 days before it peaks. And so when you get that cold virus, you feel miserable for a couple days, right? You first get it, then it takes enough days for it to build up where it causes effects in your body, but it being in your body causes your body to build up a defense. And part of that building up of defense is building memory cells, so that when you come in contact with it a second time, you have those memory cells that can produce a stronger and faster response, typically before you feel symptoms. So it dramatically reduces the effect. The goal of a vaccine, therefore, is to expose your body in a not threatening way so that you don't get sick the first time. but you still get that exposure, primarily to the proteins, right? Some use dead viruses, some use the proteins from dead viruses, and now more and more we're seeing these messenger RNAs where we make the proteins ourselves. The point, though, is to present that to the body so that the body can recognize it being foreign and build up a defense. Sometimes that building up of a defense is your body responding to the way it would be when you were sick. So it can sometimes cause fevers. It can sometimes cause achiness and things like that as a result of it, not because you're getting sick, but because your body is producing an immune response so that it can build up a memory. All right. So from some people, the side effects are more minimal, but in others, you get that response like you were sick because that's what you need to get to build up that defense. The difference is the virus isn't there. So while you may have a fever, you may feel achy for a day or two, it's not nearly as bad as truly being sick because you're not being sick because there's nothing actually attacking your body.
So this kind of, you're reminding me of the argument of parents who want to vaccinate their kids and parents who don't want to vaccinate their kids because of what happens with the vaccine when the kids are vaccinated and the side effects that could possibly happen.
What side effects?
So
So you mean like the fact that your arm is sore and you may have a slight fever, you might feel achy?
Well, I know that some people say like,
how does okay, but how does that person feel when they actually get the flu instead of the flu vaccine?
I don't agree with it. It brought up the argument of the whole, I don't want to vaccinate my kid because they could get some kind of, I don't agree with it, but that's like, kind of like, that's what they're afraid of, right? Is like the getting it and having that reaction and then, but it's not really getting it.
What I think, so again, I can't speak for anti-vaxxers, but I find that most of the arguments that I've seen fall in one of three categories. One of them, as Amanda pointed out, is that some may contain some level of things like mercury, right? However, I would point out to you that you get more mercury from eating a banana than you do from a vaccine. And not every vaccine has mercury in it. Right. So, but the few that do have such minuscule amounts that you get more mercury from eating a banana than you do from that vaccine. So it's those types of things, heavy metals or other things that may be in there that way. Others are, and again, we're not even talking about the nut jobs that think, that Bill Gates is putting a microchip inside of you or something along those lines. The other big argument that I see has to do, and things like eggs or things like that that cause allergic reactions. And again, those types of things have been in previous ones, but are no longer used in those in almost all vaccines. There are one or two that do still use eggs and things like that, but they're aware of that and they're careful about that. Yes, there are people who believe that it makes you sick. And I find there's two reasons for that. One is because, as we just talked about, it causes people to produce an immune response. But the other problem that I think people don't realize is the second you walk out the door with that shot, you are not suddenly and instantly immune from that illness. It takes about a week or to 10 days to build up that defense. So if you get sick in that first week, then you're still going to experience symptoms of being sick because you're not protected from it yet, right? Or here's the other thing that happens for some people, right? Especially like someone who got a flu vaccine now. And again, I don't mean in normal time. I mean in normal times, not like in the COVID world that we're in here. Well, we'll get to that one in a second. But in normal times, the flu season is months and months long, and it may be 3 months in when someone finally decides to get the flu shot. Well, what was the motivation to get the flu shot? Maybe they had been exposed to somebody who was sick, or they flew on an airplane and so, maybe I should get my flu shot when I get back or something along those lines. A lot of times they're motivated by they were in an experience where they may have been exposed. And if you've already been exposed and already been sick, getting the flu shot's not going to help you. So either people get sick just before or just after the flu shot, in which case it's not the flu shot that got them sick, they just got sick and it happened, right? Correlation does not mean causation. Lastly, let's talk about the autism thing. There was one study 20 some years ago where an author came out claiming to have a correlation, a causation, or really even he just had a correlation between autism and vaccination. That study has been 100% disproven. The author of that has come out and admitted that he lied, and he has been disbarred and has been denied the ability to practice medicine ever again as a result of that. And yet still 20 some years later, this is still an issue that has to be brought up and has to be discussed. There is 100% no correlation between autism and vaccinations.
And another kind of roundabout thing.
Yes.
Say that the COVID vaccine just came out. So you had COVID, you get the vaccine. Does it still do the same thing, or would it react differently?
So there's the question, the big question about that, and again, this is one of the questions that is still that is one of the things I love about what's going on right now, but is so incredibly frustrating, is because of the immediacy of the news, the immediacy of social media, the availability of information. So many people for the first time are experiencing real science. This is what real science is. Real science is that where we learn information, we gain information, we change our perspective based on that information, and we continue to add knowledge and continue to modify our beliefs based on what we find. One of the things that made me want to pull out what was left of my hair was to hear people from the previous administration, harping on Fauci about how 6 months ago he was saying A, and now 6 months later he's saying B. Well, that's because he's not some stuck in the mud idiot who just decides that he's going to have beliefs A, B, and C and doesn't change it no matter what, right? He is a scientist. And as we gain more information, we change our understanding, we change our perspective. One of the interesting things about this virus is we don't really understand the full term implications. We don't know when you get sick, how long that protection, how long those memory cells last. Some memory cells we have last longer than others. So if you got COVID at the beginning of this a year ago, you may or may not have a robust immune response for that still. So getting the vaccine could potentially be something that would be helpful. It's one of the debates going on with many healthcare professionals right now. Many healthcare professionals are foregoing the vaccine, not because they don't believe in it, but because they don't want to take one away from somebody else because they had it 6 months ago, and so they don't want to take it from somebody else. So we don't know how long it lasts for. Assuming you still have that robust memory, then taking the vaccine certainly wouldn't hurt, but it's really not going to change anything significantly. If you've lost those memory cells because you had COVID a year ago, then having it now would act like a booster, the same way you get a tetanus booster or you get a measles booster or something along those lines, where it could help to strengthen what you have. Now I see there's a bunch of text writing going on. Hold on, let me read this. Yeah, again, one of the I don't get very political in general, but definitely in the class. But I'm a very strong believer in science. I'm a very strong believer in medicine, and so it's been very frustrating for the past 4 years to have, to talk about alternate facts, to talk about the distrust of science and medicine. There was nothing that thrilled me more to see that Biden, one of the first things Biden did in office is he is promoting the Department of Science and Technology to an actual cabinet position. So the person who's in charge of that is now going to actually be a member of the president's cabinet, because it shows the importance of science and the importance of technology in today's world. And to deny that just is frustrating. Well, I wouldn't say that necessarily the antibodies are better than COVID. I haven't seen those studies, so I can't speak to that. what I can say is that it definitely reduces the severity of the COVID for people who are exposed to it. One of the other big questions is if you get the vaccine, it stops you from having a vigorous response, but that doesn't mean you're not still making the virus. And so you still could be potentially able to pass it as well. There was a lot of talk, remember, 6-8 months ago about the silent spread of it from people who are asymptomatic. Well, we may have a similar issue with people who are vaccinated and so aren't showing symptoms, but are still able to spread it. So again, from a theoretical standpoint, it is very interesting to watch science at work. It's terrifying to think of it in real life of what's going on and what we're dealing with in this situation. And it's frustrating to see people who don't understand it as they're working through it, but it is all good things. All right, that took us off way on a tangent. But again, I think that in these times, I think that is definitely, those were good questions to ask. And I think it was worth spending some time. And I apologize for getting on my soapbox. But like I said, I try not to get too political, but science is something that I'm passionate about. And this is, and we're all seeing it real time right now, which is both good and bad. All right, so we have this process of elongation, where we have this DNA RNA hybrid using our complementary base pair rules. And again, anytime our DNA has a C on it, right, our RNA is going to put a G. Anytime our DNA has a G on it, our RNA is going to be a C. Anytime our DNA has a T, our RNA is going to put what again?
A.
Excellent. And anytime our DNA has an A, then our RNA is going to put a
U.
U.
There you go. So using those complementary base pair rules, we make a temporary strand of RNA. And again, the only thing I don't like about this picture is technically, as this is being made, it's not messenger RNA yet. Remember, this needs to be modified before it actually becomes messenger RNA. Now, once it latches on at that promoter region, it continues down, but it doesn't continue down forever. Remember, eventually it's going to reach that termination signal, at which point it is going to let go. Our DNA is completely unchanged by the process. We have a strand of RNA, but again, remember, it needs to be modified. And once it is modified, then we have a complete strand of messenger RNA. And as we talked about, once we have that messenger RNA, where does it go?
The rough ER.
Yeah, it's going to go to the rough endoplasmic reticulum. And again, while we're sitting here discussing, I don't have a problem with you guys using R, but remember on the exam, spelling matters. ER is an abbreviation, and abbreviation is a spelling error. So get used to, if you start saying rough endoplasmic reticulum, then you'll be more likely to write rough endoplasmic reticulum on the exam. All right, excellent. Now, we have this messenger RNA, as we talked about. And we are then going to use it for translation. But we have to go back to that original problem we were talking about. There are only 4 nucleotides in our RNA, but it needs to be able to code for 20 different amino acids. So as we talked about, nucleotide A cannot be, right, methionine, right? Or C can't be, lysine or something like that, etc. That won't work because 4 doesn't go into 20 that way. And it can't be that A stands for 5 different ones and we get to guess which one we use. So how are we going to resolve this issue?
Triplets.
Exactly. We're going to need to use a sequence of RNAs, nucleotides. And you're absolutely right, it is going to be a triplet of that. And we can figure out why that would be, because of course, if we were to do sequences of 2, like AA and AC and AG and etc., to calculate how many different options we would have 4 different bases that we are going to do 2 times. And 4 to the second power, what is that? That's 4 times 4. 16. That's not enough. However, if we do things like AAA, ACA, etc., that way, we then have 4 to the third power. And what does that equal?
64.
64. More than enough. In fact, that means that multiple 3 sequences of nucleotides can code for the exact same amino acids. And the sequence of 3 nucleotides on our messenger RNA, we call a codon. Here, we have a pretty chart for our codons. Your book has this, your lab manual has this. You can find them all over the interwebs. Do you need to memorize your 20 amino acids? No. Do you need to memorize your codon chart? So that you know, do you need to have memorized that CCU codes for the proline amino acid?
No.
No. Do you need to be able to read a chart like this?
Yup.
Absolutely. And I noticed something interesting about this chart and everyone like it. There are 4 specific codons that are highlighted. One happens to be highlighted in green. And what is the one that is highlighted in green?
Start.
Well, but which codon is it?
AUG.
AUG. But you are absolutely correct. Green usually means go, and that is indeed the start codon. Not only is it the start codon, but it also codes for a specific amino acid, and that specific amino acid is methionine. So that's one you have to memorize, and you have to memorize what it does. What are the other 3 highlighted codons?
UAA, UAG, and UGA.
Oops. Sorry, I did that backwards. UAA, UGA, and UAG. Those, of course, are red, so those must be what?
Stop.
These are the stop codons. And notice stop codons do not code for any amino acid. These 4 codons you do need to memorize. You need to know what they do. But all the rest, you just need to be able to read a chart like this, be able to use a chart like this or one similar to it, to be able to determine which particular amino acid we would need. All right? Questions on that? Sure. Could I ask, so obviously I could ask you the 4 codons you need to know and need to know the definitions of them and what they are. Absolutely. Could I give you a sequence of a messenger RNA and you would need to determine what the sequence of amino acids are? Sure, I could do something like that. Absolutely. I'd have to give you a chart like this to be able to work it out. But yeah, I could ask you a question like that. Sure.
Is it possible if you give us coding stand of DNA and tell us to find the polypeptide?
Absolutely.
Really?
Yeah, absolutely. Or I could do it the other way too. I could give you a sequence of amino acids and have you work back to the DNA.
Okay.
Or you could be nice and give us easy questions.
Well, and that's the, well, I was going to say the beauty, but it's not the beauty. I don't determine which questions you're going to get. So I don't get to give you easy or hard questions. It's all random. I've made the bank of questions, all of which I think are good questions, but whether you get ones that are slightly easier, ones that are slightly hard, I have no control over that. There isn't like a dial, like as I like a student more, I can give them, sorry, give them the easier ones. The student I don't like, give them the harder ones. It doesn't work that way. But of course I like all of you, so you would all get easy. All right, excellent. Now that we understand this, we can start talking a little bit about translation. Translation is, again, our job of converting that messenger RNA into a protein. However, messenger RNA is not the only protein we need to make this work. There are two, other nucleic acids that we need to make this work. There are two other RNAs besides messenger RNA that we are going to use in this process. The first of these is transfer RNA, which once you've written out once, you can abbreviate tRNA. It is the smallest of all of the RNAs, only made up of about 80 nucleotides. We can take a look at it. Come back to this in a second. We can take a look at it here. Here we see, notice this is the secondary structure. This secondary structure then folds up into our 3D structure. And again, I'm not going to make you, draw this or recognize this. However, usually when we draw the transfer RNAs, it is as a tube. Now they bend it. It's not as easy for me to bend it on the board here. But there are two key components to this transfer RNA. Notice at the top of the transfer RNA is a tiny little sequence where it is able to bind to an amino acid. And down here, notice at the very bottom, it has a sequence of 3 RNA nucleotides that are used to identify the RNA. And this sequence we call the anticodon. The job of the transfer RNA is to bring the correct amino acid to the correct sequence based on our RNA. Now, so far, there's only one RNA codon that I have memorized, that I know the specific amino acid associated with it. And what would that one be?
The start codon.
Yeah, and what was the start codon? AUG. AUG. AUG is the start codon. And we know it codes for methionine. So what that means is there is going to be a transfer RNA that is going to bind methionine up here. And whenever our RNA asks for it, it's going to bring it. However, to bind to know which one to bring, we have to have the correct anticodon. So using our complementary base pair rules, what must these 3 nucleotides be at the bottom of this so it will match our start codon? Well, what's complementary to A?
UAC.
There you go, UAC. So notice this particular transfer RNA with this anticodon will bring the methionine to the start codon. So our transfer RNA basically links the amino acids to the specific codons. And that's what it's going to do. So basically, we draw it as a cylinder with an anticodon at one end and a binding site for an amino acid at another. Now, I happen to have this one memorized, as do you, but let's go back. Make sure we understand this. As I mentioned, CCU is the RNA, messenger RNA codon for proline. So which transfer RNA would bring proline to the party? If the codon was CCU, what would our anticodon be?
GGA.
GGA. If our codon was AGC, what would our anticodon be?
UCG.
UCG. See how easy that is? If you know the messenger RNA codon, you know the transfer RNA anticodon. Just using those base pair rules. Someone way at the beginning of this asked me how you remember the base pair rules. One of the ways you're going to remember them is you're going to use them a lot. So that's how you'll get used to doing it. All right. Perfect. There is a third type of RNA that is needed as well. That third type of RNA is ribosomal RNA. Now, ribosomal RNA by itself doesn't do anything. The ribosomal RNA plus a bunch of proteins, well, that's how you make a ribosome. So a ribosome is basically made up of ribosomal RNA and proteins. We've seen how we need to relate the transfer RNA to the messenger RNA. Well, we need a place for that to take place, and we need a place to help to facilitate some chemical reactions to take place. And that's what our ribosome is. Our ribosome has two subunits to it, a large and a small subunit. I'll cheat and draw it here, and I'm going to draw this simple. Here is the small subunit, and on that small subunit, there is a binding site for the messenger RNA. That's way too big. We also have the large subunit. Oops. I'll make that blue again. And the large subunit has 2.5 sites in it. Since I don't have to move this one, we'll make this easy. These are 2.5 sites for transfer RNA. One of these, and they have very, very technical names to them. This first one is the A site. As it turns out, the A site is called the A site because this is where we bring in new amino acids. And amino acids start with A, so we call it the A site. The middle site is the P site because when we start sequencing amino acids together, we get polypeptides. So this holds the polypeptide strand. So it's called the P site. And the last site The half site, the third site, however you want to call it, I don't care, is known as the E site. And guess what happens in the E site?
Exit.
Exactly. The E site is where the transfer RNA exits. Hence the term E site. And if you don't like my silly little block drawing, here is a pretty picture from your textbook showing that exact same thing. So this big globular brown thing is a ribosome made up of proteins and ribosomal RNA. And its job is to bring the messenger RNA and the transfer RNA together. And that is exactly what it's going to do. All right. This process of translation is a little bit more elaborate than transcription. So I think we want to come at the information fresh. And especially since we took our little side trot into COVID, I think now is a perfect place for our first break. Let's go ahead and take our first break here. We'll take a 15-minute break. We will restart at 1:45, and at 1:45, I will start the recording. I agree. At that point. All right. Questions on that? So
Could you go to the translation slide?
I'm sorry?
Could you go back to the translation slide?
That? Yeah.
Thank you so much.
No worries. And again, remember, all these slides are on Canvas, so don't feel that you have to furiously write down everything that's on here. Because remember, you've got all of these ahead of time. So you can look at these, print these out if you want, but you have these, so don't feel pressure to write this stuff down. All right, any other questions? All right, see you guys in 15 minutes. All righty, so again, our goal now is to talk about translation. And again, our goal here is to convert our strand of nucleic acids into a strand of amino acids. And as I mentioned, we need 3 things to be able to do this. First thing we need is a strand of messenger RNA. So let's start with that first. Oops, let's do it this way. All right, so this is our messenger RNA. Oops, no, I want that there. If I was clever, I would have set this up during the break, but apparently I'm not clever. There's our messenger RNA. We also need our ribosome. Our ribosome, we know we have 2 subunits to. So I will make this my small subunit. And then for my large subunit, I'm going to cheat and draw it. so that I can put, oh, I didn't do this long enough, did I? 3, 1, 2, 3, 1, 2, 3. No, I did do this. Oh, I did that wrong. Shucks, got to do that again. All right, let's cheat and do this part first. 1, 2, 3. All right, that got a little wonky at the end, but someone remind me again, what is this first binding site called?
The exit.
Site.
E site, absolutely.
Oh.
What is the middle one called?
The P site.
P site. And what is the first one called?
A.
A site. Excellent. So there you go. We have, and again, remember this has a binding site for our messenger RNA. I'm not going to bother drawing that, but we have that there. And we need some type of transfer RNA. So we'll put a transfer RNA. Whoops, no, I want green. That we know is going to have a binding site for an amino acid on top. And some anti-codon down here. All right, so these are the 3 things we need. We need our transfer RNA, our ribosome, and our messenger RNA. These are the 3 things we need to come together. Now, we also need a place for them to come together. So where would be a convenient place to start translation at? Not a trick question. How about the start codon? Absolutely, AUG. And so that's exactly what's going to happen. Our small subunit is going to start working down our messenger RNA until it finds that start codon. And when it finds that start codon, it is going to lock on at that start codon. Once our small subunit does that, then our large subunit is going to come and lock on to that as well. I guess I have to move those for right now. And the third thing that's going to happen is our transfer RNA is going to come in and bind as well. Now, it can't be any old transfer RNA. It can only be the one specific to the codon. So what would our anti-codon have to be in this location?
UAC.
UAC.
Oops, that I think is too big. 24. Let's try that. I think that's too small, but it will make it work. I'm sorry, UAC. Excellent. So that's the one with our anti-codon that's going to come in here and matches our codon. Perfect. And which amino acid is it going to bring in with it?
Methionine.
Methionine, absolutely.
What was the green in the middle of the block? Was it a protein?
No, remember the transfer RNA is messenger RNA.
Transfer RNA. Okay,
So
thank you.
this is just the symbol of that transfer RNA, that clover leaf shape that folds up basically into a cylinder for that. And as we mentioned, this is going to have methionine in it attached to it. Excellent. So these 4 things all come together. The messenger RNA, our ribosome, both the large and the small subunit, and our transfer RNA. With the specific anti-codon, all come together. These 4 things all come together. Messenger RNA, small subunit of the ribosome, large subunit of the ribosome, transfer RNA. Those 4 things all come together, and they come together at the start codon. Boom, that's step one, right? Because Amanda, we know, again, there's only one, well, there's 4 codons we need to know, the 3 stop codons and the 1 start codon. The 3 start codons, remember, as we talked about, do not code for amino acids. But we learned that the start codon codes for, not only is it the start, but it codes for methionine. So not only is it the first place we begin, but anytime we're going to use methionine within this, it's going to use this particular UAG codon. So that's the only one you have to memorize. You have to know the start codon codes for methionine. All right. So again, these 4 things come together. But notice there's still an open A site. So once these 4 things come together, then a second transfer RNA is able to come in and bind in the A unit. Now, again, can it be any old transfer RNA that's going to be able to come into here?
No, it has to be specific to the.
It has to be specific
yeah,
to the codon. It has to have the specific anti-codon that goes with the codon. And what would the specific anti-codon here be?
GGU.
GGU.
Excellent. And this transfer RNA is going to bring with it an amino acid. Now, do we happen to have memorized which amino acid this is?
No.
No, anybody have that memorized? No, I don't either. But we'll just name it amino acid 2. Now, if we cared enough, we could go back to that chart and figure it out. But guess what? We don't care. We just know that it's the second amino acid. All right. So first thing that happens is the 4 things come together. Then a new transfer RNA bringing a new amino acid comes in. At this point, everything is filled. So at this point, now our ribosome is going to do its work. And what that, I'll write this down there so it's out of the way. So let's say it this way. When both A and P sites are filled, our ribosome does its work. And its work is twofold. The first thing it is going to do, great question. Great question. When we make a strand of amino acids, when we make this polypeptide, absolutely every single time the very first amino acid is methionine. No, does that mean every single functional protein in your body, methionine is the very first amino acid? No, because when this strand of amino acids leaves the ribosome, where does it go? When our strand of amino acids leaves the rough endoplasmic reticulum, where does it
Golgi,
go?
would it go to the Golgi body?
To the Golgi body, absolutely. And when it goes to the Golgi body, what does the Golgi body do to it?
Modifies it.
Modifies it, cuts it, it folds it, it puts the pieces together to make a functional protein. One of the things that allows it to fold up properly is it cuts off the beginning. When it cuts off the beginning, it helps to allow that amino acid sequence to fold up. So when we make them, they always start with methionine. But once we get to the Golgi apparatus, it's going to lop off the first couple amino acids, and then that's going to fold it up into pieces. So every functional protein in your body doesn't start with methionine. But when we make it, yep, every single one is going to start with methionine. Which is why we bother to memorize it, because we always know it's the very first amino acid that's brought to the party. All right. Excellent questions. Any others? All right. Let's talk about the ribosome's work. The ribosome does 2 things. The first thing it's going to do is make a bond. right? This, of course, is going to be a bond between our amino acids. So it's going to form a peptide bond. And anybody remember that process of how we form bonds? What was that process of bond formation called?
Anabolic reaction.
True, it is a anabolic versus catabolic type of reaction, true, but that wasn't the terms we used when we were talking chemistry. When we were talking, for instance, like in our carbohydrates, when we put it together.
Hydrolysis.
Well, hydrolysis, remembers when we broke a bond, we used water to break a bond. So how do we make a bond? Remember, when we make a bond, a water comes out. So what was that process called?
Dehydration.
Yeah, by dehydration synthesis. So we are going to make a bond using dehydration synthesis. Of course, as a result of that, as we just mentioned, what do we get as a result of that? We get a water. And we can then use that water for step 2 to make a bond. Pardon me, to break a bond. And of course, we are going to break that bond between the amino acid in the P site and the transfer RNA. And we're going to do that, by hydrolysis. So, step 1. We make a bond between the 2 amino acids. When we make that bond between the 2 amino acids, as a result of that, we pull out of it a water. That water that comes out of it as a result can then be used to break this bond between the first transfer RNA and our first amino acid. So our ribosome facilitates these 2 chemical reactions. We make a bond and then we break a bond. At this point, we've done all the work we can do at this site. So the next thing we have to do, right, like any good Friday night, what do? You sit on your phone and you swipe right. And that's what we're going to do here. This whole ribosome shifts to the right on the messenger RNA. The large subunit shifts to the right. The small subunit shifts to the right. Notice when it shifts to the right, 3 things happen. Our first transfer RNA, which was in the A site, is now in the E site. And now that's in the E site and it's not connected to the methionine anymore, what can it do? It's not a trick question. What do you think it does in the E site?
Exit.
Exits. There you go. Absolutely. So it exits.
Isn't it still attached to the secondary amino acid?
Yeah, it's still, yeah, because we made that bond. So that bond is holding it together. Remember, our whole point is to make a sequence of amino acids. Right now, I've got a sequence of amino acids. Not a very long sequence of amino acids, but it's a sequence of amino acids. Notice the second thing that has happened is the transfer RNA that was in the A site is now in the P site. And the third thing that has happened is now we have an open, oops, don't want to do that. We now have an open A site. And guess what can fill this A site?
A new protein that will carry a new amino acid?
Not a
with-
protein. Remember, these are not proteins. The transfer RNAs are RNAs. They're strands of ribonucleic acid. This is not a protein. This is a RNA. It's a transfer RNA, but you have the right idea. A new transfer RNA is going to come in. Again, it can't be any old transfer RNA. It has to be the one with the specific anticodon. And what would our anticodon here be?
AGG.
AGG. Excellent. And it is going to be bringing in a third amino acid. Do we happen to have that one memorized?
No.
Nope. And do we care? Nope. Amino acid 3. Whoops. Excellent. Everything is locked and loaded. Oops. Everything is locked and loaded now. There you go. Glycine. Perfect. Excellent. And again, we can absolutely look it up. We don't care. So everything is locked and loaded. So now the ribosome does its work. What is the work of the ribosome? It's the first thing it's going to do.
To make the bond
It makes a bond. Where?
between amino acid 2 and amino acid 3.
Excellent. Whoops. I want that to be orange. Excellent. What process does it use to do that?
Dehydration synthesis.
And as a result, what do we get from that process?
Water.
Excellent. That water can then be used for our second process. What's the second process?
To break the bond
Where?
between amino acid 2 and the GGU.
Exactly. In the P site between the transfer RNA and the second amino acid here in the P site, we break that bond. So again, hydrolysis to break the bond using the water we got from the dehydration synthesis. And yes, well, again, this is happening at the ribosome, but yes, most of the ribosomes are attached to the rough endoplasmic reticulum. So yes, mostly this is happening at the rough endoplasmic reticulum. Excellent. We have now made a bond and broken a bond. So what do we do next?
Move everything to the right.
There you go, swipe right. Oops. There. Wait, oh, hold on. No, I was right before. There, perfect. Excellent. The transfer RNA in the P site, what happens to it? It exits. Oh,
Exits.
assuming I can grab it. Let's hope that I can. There we go. So it leaves, of course, with its anticodon and its stem. Whoops. Grab the wrong part. Oh, grab that stem too. There we go. Perfect. Now notice these transfer RNAs, once they leave, they can go and be refilled again. Ugh, come on. Too many Christmas. There we go. All right, these can go and they can get a new amino acid put on them. This can get another methionine put on top. This can get another one, and they could actually be used multiple times in this same sequence. But it leaves. Our third transfer RNA is now in the P spot.
A site.
And the A site is now open. So I've run out of shades of green, so I'll just go back to the original one. A new transfer RNA comes in here. And again, can't be any old transfer RNA. It's got to have the right anticodon. What would the anticodon here be?
UGU.
U. Excellent. And again, it's going to bring an amino acid. We don't have to have it memorized. That is okay. We just know that this is now the fourth amino acid. What happens next?
bonds break?
Do we break the bond first?
Make the bond.
No, we make it.
Make the
and then we
bond.
break it.
Make the bond first using dehydration synthesis. That gives us our water. And then once we get our water, we can then use that water to do what?
Break the bond by hydrolysis.
Yep, using hydrolysis, break the bond between the transfer RNA and the amino acid in the P site. And once this is done, what happens next?
Shift.
Shift. Oops. It all is going to shift to the right. Absolutely. Yep, I keep forgetting to shift my small subunit with it. Sorry. Now, remember, a typical polypeptide is going to be thousands of amino acids long. So this process is going to happen 1,000 times until finally something different happens. And what's that different thing that's going to occur?
Stop codon.
We're going to run into a stop codon. What were the 3 possible stop codons again?
UAA, UGA.
UGA,
And UAG.
UAG,
UAG, and
UAA.
UAA. Have we happened to run into one of those?
Well, yeah, once we go to the right. Oh, wait.
Yeah.
Oh, yeah.
I shifted to the right. So, yep, we're there. Absolutely. Yep. So, absolutely. Now, remember, we said these were the stop codons. And remember, we also said they don't code for an amino acid. What these special transfer RNAs do is something slightly different. This transfer RNA, when it comes in, and again, it's not any old random transfer RNA, it's got to have the correct anticodon. What would the correct anticodon be?
ACU.
Excellent. So here is our, I'm going to shorten that. Here is our stop codon with the ACU anticodon matching up with the start codon. And what happens is this transfer RNA doesn't bring an amino acid. What this transfer RNA does is it brings a water. What might that water be useful for?
Breaking the bond.
Yeah, water is used to break bonds, exactly. And so this water is used to break this bond between our remaining transfer RNA and our amino acid sequence. Notice when this breaks the bond, is there anything holding this together anymore?
No.
No. And so what's going to happen is our small subunit, our large subunit, the transfer RNAs are all going to go away. And what we're left with is our completed sequence of amino acids. And this completed sequence of amino acids, as you guys just pointed out, is going to go to the Golgi apparatus to be modified and folded into a fully functional protein. Questions on this process?
So all of this copying was not done on the DNA, it was done on the messenger RNA. Where did the messenger RNA come from again?
Remember, the messenger RNA is what we made in transcription. In transcription, we made the messenger RNA. And then here in translation, we converted that messenger RNA into a sequence of amino acids.
Oh, okay.
So the red transfer RNA is associated with the stop codon, and it brings water. Yes, that is correct. Again, I did the best as I could drawing this on the board. We've gone over this together. Let's do the entire process again with all the pretty pictures and all the pretty words from your textbook. Now, this actually isn't from your textbook. This is a very, very simplistic illustration of this. Although your book, I will say, has an in focus that is really, really excellent and does an excellent, excellent job of showing this. The two ways that this illustration simplifies things, and again, it doesn't hurt our understanding of it, but the two ways it does this is, notice for starters, it does, oh, I don't know why I'm using white here. It doesn't show us any of the sequences before the start codon. There will be nucleotides before the start codon, and there'll be nucleotides after the end, the stop codon. So it just shows us the functional part of the messenger RNA. No, there will be stuff before and after it. And notice they didn't bother giving us an exit site. They just focused on the A site and the P site because that's where all the work is taking place. So again, neither of those things affect how this process works, but I just wanted to show how they're simplifying it a little bit. So the very first thing that happens when that messenger RNA gets to the rough endoplasmic reticulum is that 4 things come together. Great question. Nothing. That stuff before and after the start and stop codons isn't used. It's non-functional. It's just buffer at the end so that nothing happens to it. All right. All right. So 4 things come together. Our small subunit, our large subunit, the messenger RNA, and our transfer RNA. And they all come together around our start codon. No, great question. So actually, DNA, they think some of that repetitive DNA that I talked about at the end could be those kind of end caps at the end. I was going to save this discussion for the end, but notice, let's go back to my illustration and we'll do this here. With this strand of messenger RNA, notice, was our messenger RNA changed, modified, damaged at all by this process of reading it and making a single sequence of amino acids?
No.
No.
However, if I need a protein, do you really think I just need 1 copy of this strand of amino acids?
No.
No. So the advantage of this messenger RNA is it can be read many times. In fact, what's really cool about it is because of the way the ribosomes work, while one ribosome grabs on and starts moving down, a second one can grab on and start moving down. So multiple ribosomes can actually be reading the DNA at the same time. And this allows us to make many, many copies of this sequence of amino acids. However, once we start making a sequence of amino acids, do we want to keep making that sequence of amino acids forever? No. So one of the things the cell's doing at the same time, while we have those ribosomes running down the ends, making the proteins, we have some enzymes that are coming through and lopping off the very first nucleotide. And then it'll lop off the second one. And then a little bit later, it lops off the third and the fourth and the fifth and the sixth and so on and so on and so on and so on until eventually it gets to the meaningful ones. Once it lops off that A, can this messenger RNA be read anymore and be used to make a protein? No. So the strands at the beginning limit how long we can read this for. The more nucleotides it has before the start, the longer it's going to be stable, the more proteins we make. If there's only one, then once we lop off those first two, it's no longer functional. We're only going to get a very small amount of proteins. If there's dozens, we're going to get a large amount of the proteins. So it does affect how much of the proteins we make or how long we can make those proteins. I'm not sure what you mean by the buffer code. Enzymes, there are enzymes that don't care what the first nucleotide is. They just lop off the first nucleotide. Doesn't matter what letter it is. And so then another one will come along a little bit later and lop off the next first one. And then another one will come and lop off the next one. And then another one. And again, these are random events that are occurring. But every time one of these enzymes ends up by the RNA, it's going to lop one off until eventually it's not functional anymore. Yeah, enzymes are breaking down the messenger RNA, breaking the bonds of the messenger RNA. Notice, remember, the ribosome is made up of ribosomal RNA and proteins. Guess what the proteins are? Primarily enzymes. They give their structural proteins to give it its shape. And there's some enzymes that facilitate those chemical reactions, the making bond and the the making of the bonds and the breaking of the bonds that we're talking about. All right, so back to the pretty pictures. 4 things have to come together: messenger RNA, transfer RNA, small subunit of the ribosome, large subunit of the ribosome. All 4 of those things come together, and they all come together on the start codon. And again, that start codon tells us which transfer RNA can come in. It's got to be the one with the correct anticodon. And of course, the correct anticodon for UAG is UAC. AUG, sorry, is UAC. And as we know, what is that first amino acid that's going to be brought to the party?
Methionine.
Methionine, there you go, methionine. Excellent. So yes, it is a transfer RNA that is binding to the stop codon in the messenger RNA. It's just bringing a water. So when they talk about it being a release factor, they're really just saying that it brings a water that we can then use for hydrolysis. Not a protein, transfer RNA. All right. Once we have our first transfer RNA in place, our A site is open, so a second transfer RNA can come in. That second transfer RNA that comes in, again, can't be any old transfer RNA, has to have the specific anticodon for the codon. And it locks into the A site. Once we have got this site filled with all the transfer RNAs that we can, all 2 of them, this is when the ribosome does its work. Make a bond and break a bond. First, we are going to form that peptide bond between the 2 amino acids. That, of course, produces a water, and that water can be used to break the bond between the first transfer RNA and our first amino acid, which, as we know, when it shifts, is going to enter the E site and exit. And so the whole ribosome shifts to the right. Our first transfer RNA exits. Our second is now in the P site. And now our A site is once again open for a new transfer RNA to come in using a specific anticodon. Once it's in, we make a bond and we break a bond. And again, this process continues literally thousands of times until something different happens. That something different that happens, oops, is a stop codon. When that stop codon comes in, it brings a water with it. That breaks the final bond and the whole thing disassembles. Again, didn't modify or change our messenger RNA. Yes, ATP is used in this process. When you take micro, if you take 400, they may get more into all of the steps of what the ribosome is doing. We're not going to worry about all of that process. But yes, absolutely ATP is used in this process. Now, as I promised, your textbook's got a great illustration that does a really nice job of showing this, the in focus that it has, where again, it shows our 3 sites in the large subunit of our ribosome, the transfer RNAs, bringing in the new one, forming that bond, breaking that bond, shifting down as it goes, so on and so forth, until we reach the stop factor, the stop codon, which it calls a release factor. But again, all that release factor is doing is bringing a water to the party. And that water then allows it to be released. Notice it even shows the enzyme that's putting the amino acids on our transfer RNAs. Now, as I mentioned, here's a pretty illustration showing how a single messenger RNA can be being read by multiple ribosomes at the same time, at which point you may say it can't really look like this. We've covered far too much information for this to just be this topic just being the exam. And again, you knew we were talking about this today, so there's no reason you shouldn't have been looking at it this weekend. Here, using an electron microscope, we can actually see a single strand of messenger RNA where there is over 10, close to a dozen different ribosomes all reading it at the same time. The other thing to remember is this isn't that tricky. While it may be a long process, it isn't a hard process. Basically, you are taking a chunk of DNA with those nucleotides of that coding strand, probably the template strand. Using that template strand, you're coming up with a strand of messenger RNA with codons. Those codons correspond with an anticodon, which corresponds with an amino acid. Not only do you need to be able to describe this process, but as someone pointed out, I should easily be able to give you a strand of DNA. And as long as I gave you one of those charts, you should, even without the chart, you could give me the sequence of transfer RNAs. But if I gave you some chart, you should be able to tell me which amino acids are here. Or I could do it the other way as well. I could give you a sequence of amino acids, and you should be able to give me the template strand of DNA that would be responsible for it, as long as I gave you a table to be able to figure out your codons and anticodons or transfer RNAs and amino acids on. Pretty basic steps. All right, now, one more important thing. Here is the one issue that we have. The DNA in all your cells, your toenail cells, your eyeball cells, your liver cells, is that DNA the same or is that DNA different?
Different.
Different?
Same?
Same? Okay, I've heard one of each. Which one is it? Is it different or same? Two more sames, one more
I think
different.
it's same because the different parts of your body just use a different part of the DNA, no?
There you go. Every single cell in your body has the exact same DNA. But you are also 100% correct that not every cell makes the same proteins. Toenail cells make toenail proteins. Eyeball cells make eyeball proteins. Liver cells make liver proteins. So every cell has the information to make every single protein of your body, but every cell doesn't make every protein. And it also only makes proteins when you need them. So how do I tell toenail cells how and which proteins to make or when to make them, and liver cells which proteins to make and when to make them, and eyeball cells which protein to make and when to make them? How do I regulate this whole process? How do I tell cells which protein to make and when to make those proteins? How is this process regulated? You're right, the process is gene expression, but how do I get the cells to do that? How do I get each cell to express the genes that I want them to express? How does that work?
Is it like activating or inactivating some?
True, we need to activate and inactivate things. We need to be able to turn things on and turn things off. We almost, you could say, need to be able to communicate with the cells to be able to tell the cells what to do. Didn't we talk about body systems, organ systems that were specialized for communication?
Mm-hmm.
Which ones?
The nervous system.
And
Endocrine.
endocrine. Which one is it going to be easier for us to use to communicate to every single cell of our body to tell them which proteins to make? Do we have nerves that go to every single cell of our body to tell every single cell what to do constantly?
Wouldn't it be through like hormones in the endocrine system?
Absolutely. That is exactly how. Someone used the magic term gene expression. Gene expression is really just a fancy way of saying what proteins we're going to make and when to make them. That gene expression is indeed controlled by hormones, by our endocrine system. We release hormones into our blood, and once it gets into the blood, it can go potentially to every single cell of our body so that we can tell toenail cells to do toenail things, eyeball cells to do eyeball things. Protein synthesis, which proteins a cell makes and when it makes them, is controlled by our hormones. All right. Questions on that? Nope. No, the nervous system does very important things in our body, but expressing our genes, making proteins is not one of the things that our nervous system does. So this one's solely just the endocrine. All righty. With that, we are done with protein synthesis. That is our last major physiological process we need to talk about. However, being the smart, sophisticated students that you are, as I mentioned, when we have a process like protein synthesis that has two major processes in it, like transcription and translation, we have three possible essay questions. Describe transcription, describe translation, or compare the two. Now, if I ask you to compare the two, does that mean describe the entire process of transcription and describe the entire process of translation?
No.
No. If I ask you to compare me and my daughter, and you say she has blue eyes and I have no hair, is that a direct comparison of the two of us?
No.
Right. No, exactly. So when we're doing comparisons, and again, you don't have to set it up this way, but I often find doing it as a table is something that is useful, because then what it allows you to do to make sure you're comparing apples to apples and oranges to oranges is you can think in terms of, all right, what is the characteristic that I am comparing between the two? Excellent. Read and math heavy is not so bad. All right, excellent. So someone give me a way that transcription and translation are different from each other. Okay, excellent. Location, perfect. That's a great way. How are they different? Where does transcription occur?
Nucleus.
In the nucleus.
Excellent. Where does translation occur?
In the ribosomes.
All right. Well, I would say at the ribosome. Where are those ribosomes located?
Rough endoplasmic reticulum.
Yeah, you could say rough endoplasmic reticulum. Heck, you could also say in the cytoplasm. That would be different from in the nucleus. That would be an acceptable answer as well, if I spelled it right. What's another way we can compare them? Nope, I just got a warning. My interweb is unstable. Can you guys still hear me?
Yes.
Yeah.
Okay, excellent. All right.
Hear you.
Excellent. All right. So give me another way that they're different.
In
Function.
All right. What do you mean by function?
So like what it
One of
does.
them.
So like one is translation is what it translates or changes the language, while transcription is just does the portable copy of the information.
Okay, so I like that. So kind of differences in the function of the languages. You're right. Transcriptions, basically, it's the same. We are going nucleic acids to nucleic acids. Whereas in translation, they're different. We are going from, wow, I'm getting really bad at my spelling, from nucleic acids to amino acids. Excellent. Give me at least one more.
The proteins used.
Okay, we want to be careful. Proteins / structures that are used, right? Because what is the primary protein involved in transcription?
RNA polymerase.
Yeah, RNA polymerase, excellent. And RNA polymerase absolutely is a protein. What are the structures used in translation?
You mean which enzymes?
Well, so you get the right idea. One of the big ones is a ribosome. The ribosomes, which are proteins and ribosomal RNA. What's the other structure used here as well?
tRNAs.
Yeah, transfer RNAs. And of course, on a real exam, you'd be writing these all out, which of course are RNAs. So again, so it's not all proteins, but you get the right idea. These are the structures that are used in the processing of them, absolutely. Towards that same end, and it kind of goes along with locations, what's the end product of each of these? What's the end product of transcription?
RNA
Be more
or
specific.
mRNA.
Be more specific. Messenger RNA. Excellent. And then what's the end product of translation?
Protein sequence. Or
Right, although we
amino
want to be
acid
careful.
sequence.
It's not an amino acid sequence would be more appropriate. Or you could say a polypeptide strand. All right. So there you go. Notice I'm not describing the processes and I'm just not throwing out random facts. I am specifically comparing different characteristics between the two. When I ask you for a comparison type of question, this is the type of answer that I'm looking for. I don't want you to describe both processes. Don't just give me random facts about the processes, compare them, all right? And so, like I said, you're not required to give me the characteristics. I just like to set it up this way because it helps me to make sure that I'm comparing apples to apples and oranges to oranges when I'm doing this. Okay, so this would be the kind of thing that I would be looking for on that type of a question. And I guarantee you that these three questions, describing transcription, describing translation, and comparing the two of them, are questions that are in your test bank. Do I know if you will get one of those and which one you'll get? Nope, I have no idea because it is completely random. But I can tell you with 100% certainty, all three of those questions are in the test pool. So they are certainly questions you could have. All right.
As another difference, can we say that in transcription, the template strand is DNA and in translation, it's mRNA?
Sure, absolutely. Again, these are not the only 4 comparisons. There are probably dozens we can do. And absolutely, that's a great one, right? So you could think of the template or the starting point, right? We start with DNA. We start with messenger RNA for translation. Absolutely, another perfect example of a comparison. Yep, exactly. All right, questions on that? Excellent. So what we have now done is completed all of the information you were responsible for this first exam. And you could argue that last question was even kind of the beginning of our review, but we've covered a lot of material today. So what I want to do is I want to do 2 things. I want to take a break. And again, I'm not providing any new information at this point. So anybody who wants to leave is welcome to leave at this point. However, when we come back from the break, anybody who is interested is welcome to ask questions and we can work together to come up with those answers to try to help you to be successful on the exam. So again, you're not required to stick around. That's the other advantage of a break, is someone can leave quietly without being noticed by everybody else. But we'll come back. Sure, why not? And we can do that after the break. So let's go ahead and take one last break. I guess we'll bring up the whiteboard for lack of anything better. And then we will take a quick 15-minute break, come back at 2:55. And at 2:55, we will restart with our Q&A review. And I'll remember to start the recording at that point. Oops, God, wow. Sorry for my spelling. I should not have had so much vodka this morning for breakfast, so I apologize for that. It seems to be particularly bad today. All right, there you go.
Yeah, me neither.
Excellent. I will meet you guys back here at 2:55, and at that point, we will do our review. All right. No, I missed it. All righty. Here we are. So again, with the time that we have left, I'm happy to answer any questions to help clarify some of the information, either about the process of how you're going to take the exam or content related stuff to the exam or anything like that. I'm happy to provide what information I can to help you to be successful. So again, this is your point to ask me, not for me to sit here and present information. So tell me what you got.
This was probably asked like multiple times, but like with the exam questions, how long is it? Or like how much do you have to write?
So for the lab exam, it's probably somewhere between 60 and 70 questions. Each of them is going to be an image with an arrow, kind of like the practice exam we did. And so you have to write, rough endoplasmic reticulum or, left crural region or whatever it is, something along those lines. The lecture exam is going to be somewhere around 30 multiple choice questions, usually somewhere between 2 and 6 fill in the blanks, and somewhere around 8 to 12 essay questions. How much you write is really going to depend on the essay question. Essay question is going to be worth different points. And so, you know, you're going to write a different amount for different questions. If question's worth 3 points, then, you know, you don't need to write 5 paragraphs for something like that. If something is worth 10 points, you're not going to write 1 sentence for that. So it's going to be somewhere within that range. I haven't found.
Oh, okay.
So
I was just thinking of like writing like a structured like body paragraph and all that stuff.
no, and also for the essay questions, I don't care about your format. If you want to write it out as a paragraph, you are more than welcome to do that. If you want to do bullet points or something like that, that is fine as well. However you want to organize the information is fine. I don't care about the organization. I care that the information is there. Now, the one thing that I would say with that is that when people do use bullet points, one of the things they do tend to do is be more brief in their descriptions. And that is where you're going to get in trouble, one of the things that I am looking for and striving for and expecting on these is specificity of detail. Right? Don't just tell me that the ribosome binds to the messenger RNA. You need to tell me that the small subunit of the ribosome binds to the messenger RNA at the start codon, which is AUG. Right? You have to get, I want details. I want specificity. I want specifics and details being descriptive. And so sometimes when people do bullet points, they aren't necessarily as descriptive, but as long as you're descriptive, I don't care about your format.
Okay, thanks.
All right. Yep.
So for the lab, sorry.
Mhm.
For the lab exam, it's going to be based off of what we've done in the lab manual, correct? Or is
No, it's going to be, again, I think one of the misnomers about this class is this is one class, right? There is no lab class and lecture class. Lab and lecture are just different modalities for learning the information, right? Some things it makes more sense for me to stand and wave my hands. And some things, for instance, like when you're learning the bones or when you're supposed to be learning the bones, you're supposed to be holding those bones in your hand and looking at them and tasting them and doing all the other things that you can do with those bones to learn them. So they're just different modalities for learning the information. You are responsible for all of the information on all of the exams. However, as you saw from the practice lab exam, on that practice lab exam, most of the lab exam is going to be identification. What is this? What is this? What is that? However, at the same time, I could point to the Golgi apparatus on a model of a cell and ask you to identify it. And of course, you would say Golgi apparatus, but I could also point at it and ask you, what does it do? And you would have to tell me that it modifies and cuts and folds up and produces functional proteins and then ships them to their final destinations. So while it's primary identification, there is going to be some function on that. Essay questions tend to lend themselves really well to processes, right? But I could also ask you for identification of, identify and list 4 organelles and describe their functions or something like that as a possible essay question. So again, there isn't, and that's one of the reasons we have the test on the same day, because it's all covering the same material. Different modalities for learning it, different modalities for testing it, but all one class. So yes, there's no difference between the material that you're responsible for in the lab and lecture. It's all the same. It's all one class. Some things lend themselves better to lecture. And again, lecture exam, that protein synthesis isn't, I'm not going to ask you to describe protein synthesis on the lab exam, right? But could I show you a ribosome and ask you to identify what it was, from its symbol? Yeah, absolutely. So all the information on all the exams. So notice when we talked about Golgi apparatuses, we talked about rough endoplasmic reticulum, we talked about characteristics they all shared. You are right. When we talked about the peroxisomes and the lysosomes, they're just vesicles. So because they're just vesicles, there really wasn't an easy way to distinguish them. However, on the 2 models that we had, we made up a rule by convention. So on those 2 models, there are 2 types of vesicles, yellow ones and purple ones. So we made up a rule in this class based on convention that the purple vesicles were going to be what?
Lysosomes.
Lysosomes, and therefore the yellow ones are the
Peroxisomes.
yeah, peroxisomes. There you go. Exactly. So that was the rule we made up. So can I show you an electron microscopy view of a cell and point to a vesicle in that and have you figure out whether a lysosome or peroxisome? No. Is it possible to do that? Yeah, absolutely. But am I going to hold you responsible for that? No. All right. So vocabulary absolutely is one of those things that are important. So when you think about it, all of our organelles are made up of components, right? You have a chair and it's made up of components. It's got wheels, it's got legs, it's got arms, it's got a back, and they collectively make the chair. So we can do the same thing with an organelle, right? If I gave you an organelle, like let's start simple, centrosome, that is the organelle. That doesn't have to be that large. What were the structures that made up the centrosome?
Centrosome matrix and
Centriole.
Yeah, there you go. The centrioles and the centrosome matrix. Perfect. Right? Those are the structures. If I ask for the rough endoplasmic reticulum, what are the structures that it is comprised of?
Ribosomes and
The cisterna. Oh, sorry. Never mind.
Yeah, no, you're absolutely right.
Okay.
Cisternae, right? Those flattened membranous sacs, right? This is different from the smooth endoplasmic reticulum, which is comprised of what?
No ribosomes.
Yeah,
and
so basically
the
just
cisternae.
cisternae. Well, then I have a question. What is the Golgi apparatus made up of? Or the Golgi complex, Golgi body?
It's also made up of cisternae.
So how do we tell the smooth endoplasmic reticulum from the Golgi apparatus?
Is
the folds.
Apparatus has the vesicles surrounding.
Okay, excellent. Perfect. So one way is this is going to be surrounded by vesicles. Was there another way?
yeah, in Golgi apparatus, those they're not attached to each other. Those fold things.
There you go, the cisternae. So in the smooth endoplasmic reticulum, the rough endoplasmic reticulum, remember these are interconnected cisternae. Whereas in the Golgi apparatus, the cisternae are all separate. And it's surrounded by vesicles, absolutely. Right? So again, we have the structures making up the organelles. Those are some great examples of that, right? Exams are all about the things that we've covered in the lecture. So yes and no. You are correct in that there is far more information in the textbook than we cover in lecture. However, remember, not only are you responsible for lecture, but you're responsible for all of the homework. So the PhysioXs, the Labsters, any homework assignments, any concepts that were covered on those. Now, usually those homework assignments correlate with the information that we have covered in the lecture. So if there's a piece of information in your textbook, and chapter 2 is a perfect example of this, right? Chapter 2 on chemistry, most of that chapter is stuff that you were supposed to know coming into it. So we didn't cover any of it. So the only chemistry stuff you're responsible for is the macromolecule stuff we talked about in lecture. You're not even responsible for the stuff that was on that chemistry quiz. That chemistry quiz, remember, was information you were supposed to have coming in. That wasn't stuff we covered. So yes, you are responsible for the material we have covered some way in class, whether it was lecture or some type of an assignment or something along those lines. But yes, there is information in your textbook that we are not responsible for if we didn't cover it in any sort of fashion and way, right? Like I said, there may be some rare instances. As I mentioned, this next section coming up is a perfect example. As I said, we've got 3.5 days worth of lecture that we have to cram into 3 days. Now, the fact that we don't have a lab makes it a little bit easier, but it's still going to make for long days. So at the end of that third lecture, we all may be really exhausted. And so I may say to you, look, we're running out of time today. I don't have the opportunity to talk about burns. So make sure you read burns in your textbook because you're going to be responsible for burns on the exam, right? That would be the only instant where you'd be responsible for something we didn't cover. And I would specifically make you aware of it. But if we never talked about burns in class, you never had a homework assignment about burns, right? We didn't cover it on any assignment, then it wouldn't be on the exam. All right. What does folds mean?
I think they were talking about the Golgi apparatus.
Oh, okay. All right. All right, next question.
Sorry, go ahead.
I just have a quick one. After I write down the stuff for the smooth endoplasmic reticulum, what was the function again?
Anybody? What's the function of the smooth endoplasmic reticulum?
Detoxification, lipid synthesis.
There you go. It plays a role in synthesizing lipids, plasma membrane, stuff along those lines, but it also plays a role in detoxifying the cell as well.
Oh, okay, thanks.
Yep.
I was going to ask.
Oh, yes, and thank you. Yuli's also got it. Remember, the smooth endoplasmic reticulum in some cells can also be specialized for storage, in particular storage of calcium.
Or like muscle cells.
Yeah, muscle cells in particular is where in the ways where you'll see that. Skeletal muscle to be specific, but yes, absolutely. Go ahead.
Yeah, sorry. I've noticed that some of the functions of smooth endoplasmic reticulum and peroxisomes are similar.
Yeah.
So if it asks, for example, which organelles will liver cells have? So can we say both?
Sure, absolutely. Yeah, absolutely. That would, and that'd be a good example of one that has a large amount of both. Yep.
Okay.
All right, there's another good question here in the text. Actually, looks like there's a couple of them. So let's do this first one first. The difference between chemical force and electrical force. So remember we were talking about there are basically two driving forces on a charged particle, right? Remember, if we were just talking about this being glucose over here, then the only driving force on glucose is going to be concentration gradient, right? We have that concentration gradient where things go from a high concentration. Actually, let's just do it this way. High, where's my box? There we go. To a low concentration. Right? That is our concentration gradient. Things move from a high concentration to a low concentration. And that is true whether we're talking about glucose or whether we're talking about something like a sodium ion. Both things are going to go if there is 80% of it on this side and there is 30% of it on this side, it is going to go from the high to the low. All right. But when we are talking about charged particles like the sodium, there is also going to be an electrical force. So hold off on the new questions until I get a chance to answer these, because as you guys keep scrolling up, I keep missing them. So the electrical force, oh, and remember concentration gradient, we could also think of as the chemical force. We can kind of use those terms interchangeably. Electrical force is basically either the attraction or repulsion of a charged particle by the charge of the cell. So if a cell, for instance, is negative on the inside, then positive things are going to be attracted towards that negative, whereas negative things are going to want to move away from that negative charge, because opposites attract, and positives don't like positives, negatives don't like negatives. And obviously, the greater the charge is here, or remember, I keep saying charge, but remember, really, we're talking about membrane potential. And what did we say the normal resting membrane potential of a typical cell was again?
Negative 70 millivolts.
Negative 70 millivolts, absolutely. So normally the inside of the cell is very, very negative. So positive things really want to come in along that electrical force, where negative things really, really want to go out. So both of those driving forces, the electrical force and the chemical force, can influence which way an ion moves. However, when a cell is at rest, remember we talked about when a cell is at rest, typically it is the chemical force that is the driving force, that tends to be the larger force. So again, there will be times where it won't be the case, but in most cases, and especially now early, when we get later in the class and we talk about the nervous system, in most instances, the chemical force is greater. It has more of an effect on driving an ion and determining which way an ion moves. All right. Questions on that? Any more questions on that before we change concepts?
So with your definition or saying of the attraction or the electrical force, I'm sorry, is charged there also meaning the membrane potential?
Yes. Well, the membrane potential is a measure of the number of negative things versus positive things inside and outside of the cell. So when a cell is at rest, there's more negative things inside the cell, so the cell is more negative on the inside.
Which would make it, okay.
Yep.
Oh, that makes sense now.
Excellent. I love that. Those are words I love to hear. Excellent. All right. So any other questions on that before we move to the next question I see? Hyper and hypotonic solutions. So let's take two beakers and take two cells. And I always like using simple numbers because simple numbers are easy. Let's say that cells are 15% stuff. And I'm purposely not using the real percentages for this. So don't, and of course, 80% water. I'm purposely not using the right numbers because the numbers don't actually matter. It's the relationship of them. Let's say the cells in your body were 15% stuff and 85% water. Ideally, then, they want to be surrounded by a solution that has how much stuff in it?
85.
How much
Same.
stuff do we want in it?
15.
Yeah, we want 15% stuff. And you're right, 85% water. You're right, we want 85% water. And if it was in this type of a solution, how would we refer to this solution?
Isotonic.
Isotonic, excellent. This would be an isotonic solution. So with an isotonic solution, it's the same amount of stuff and water inside and outside. So that means, that doesn't mean water and stuff aren't moving. It means there's no net change. And so the cell is happy and the cell is at equilibrium. All right. However, if instead we took that cell and dumped it into deionized water, what percentage of stuff is there in deionized water?
0.
Yeah.
Almost.
Essentially, yeah, you got absolutely the right idea. Essentially 0% stuff, which means how much water?
100.
100% water. In this case, we don't have a balance anymore, right? And water is going to move. And which way does water move?
Inside.
Yeah, down its concentration gradient
Stuff.
or to where there's more stuff. Again, both are equally easy ways to describe that. Causing the cell to swell up, and if it swells up enough, it will burst. Right? What type of solution would we call this?
Hypo.
Hypo, there you go. Hypo means below. So hypertonic, it is less than tonicity solution. If instead we put that cell in salt water, and again, these aren't correct, but again, I'm just using fake numbers because it's simple. Let's say salt water of the ocean is 30% stuff and 70% water. What would happen in that case?
Water would come out of the cell towards the stuff.
Water goes down its concentration gradient, or water moves to where there's more stuff, and our cell would shrivel up and crenate, right? That crenation process. And this type of solution that has more stuff than a cell has inside of it, we would call a hyper. So there you go. Those are our both isotonic, but also hyper and hypotonic solutions.
So for isotonic, it was equal, right? But
Yeah,
the inside and the outside.
Exactly. And so when it's equal inside and out,
there's no net movement.
Exactly. No net movement. And everybody's happy. We're at equilibrium. Exactly. All right. AMP, ADP, and ATP. So be careful. Remember, ATP is the primary energy molecule of the cell because it's the one that has the 3 phosphates. And remember, adding that third phosphate required a massive amount of energy to make that bond. So when we rip off that phosphate, right, then we release a tremendous amount of energy, right? When you have that ADP, we still have a high energy bond there. And so when we rip off that phosphate and only have one left, it does release some energy, but not as much. So you're right, ADP and ATP are energy molecules, but ATP is really the high energy molecule. And AMP is really the building block that we start with. And yes, purple for lysosomes, yellow for peroxisomes. We did it, again, purple for peroxisome would have been easy, but remember we have that nice model where it's a difference of size. The lysosomes tend to be bigger and the purple vesicles were bigger, so we were trying to be more realistic. So that is why we use purple for the lysosome and yellow for the peroxisome. All right, excellent. I think that is all of the written questions. That's perfect. Next question.
I had a quick question. If like a region of the body has like 3 different names that are accurate, whichever one we use doesn't matter as long as it's one of the accurate is fine?
You may use any appropriate anatomical term. What region of the body are you thinking of?
Well, I was thinking the iliac / what's that called? Linguinal?
Inguinal, yeah.
Inguinal?
So what's
Yeah.
the third?
Well, just those 2. And then also some of the upper, like from the 9 plane, some of the upper one have 2, and even the middle section like has, I'm forgetting.
So inguinal, iliac, interchangeable words. The lateral aspect of your lower leg, peroneal, fibular, both of those are appropriate terms. I was trying to think of one that had 3. I don't know if there's necessarily one that has 3 that I can think of off the top of my head. But yes, as long as it's an appropriate term. Now, again, it can't be what your Uncle Billy calls it, but as long as it's an appropriate anatomical term, then yes, you may always use any appropriate anatomical term for describing things.
Okay, and then also I'm having a hard time memorizing the, not mitosis, but like the cell life cycle. Am I supposed to have that memorized? The M phase, 2G phase, S phase. Is this just as important as understanding what happens in each step of mitosis?
Yes. Knowing mitosis is important. Knowing the mitotic phase, which remember is not the same thing as mitosis, is important. Right? Because remember, the mitotic phase includes mitosis and cytokinesis. And also knowing the complete cell cycle is definitely something that is important, absolutely. Yeah, you
Okay.
need to know what's happening in interphase as well as what is happening during the mitotic phase. And remember, not every cell stays its entire life in that cycle. We also have that G0 phase, which is outside of the cell cycle, that we need to be able to talk about as well. So yeah, absolutely, we need to talk about those things.
I have a question. Wait,
Yes.
I'm trying to remember what it was. I think it was on one of the lab reviews, and it was something about the liver and what could the organelle be, but it was like it could have been lysosomes or the proxisome.
So yeah, the liver plays an important role in detoxifying. So that could be definitely something that you would want to have in there.
Yeah. So if you put one but not the other, would that still be correct, or do you need to put both since they both?
Okay. So here are the things that so here's the things that I would say. Those two are just a starting point. Obviously, most of the processing of our blood that takes place in the liver requires a lot of ATP. So it probably has a lot of mitochondria, right? We want to store nutrients. So for that, we would want probably some smooth endoplasmic reticulum, right? There's all sorts of different potential answers for that. The thing that I would remind you is twofold. One is that, again, the point of the homework assignments is I'm looking for time and effort, that you're putting time and effort into them. And so by thinking about it and having multiple potential answers, it's not so much about what's right or wrong. I'm looking for time and effort into it. However, if we take that into the realm of an essay question, if I ask you for organelles plural, should you just give me one?
No.
No. So that would be the other thing that I would say to that. If that question asks for organelles plural, then you probably should not have stopped at one.
Okay.
Okay. Looked like there's a couple quick questions on the text, so let's get to those real quickly. We're not talking about redox reactions. Again, that's chemistry. We are talking about glycolysis and dehydration synthesis, basically the ways we are making covalent bonds and breaking covalent bonds. We talked about it in making disaccharides from monosaccharides, and we've now talked about it with amino acids, where when you form that dehydration synthesis, you form the bond between an amino acids. Remember, we call that a peptide bond. So those are the two times we talked about bonding for that. All right, so we did talk about that in lecture. The other one, the question before that, so that was the easier one to answer. Let's quickly talk about the other one. When we were talking about passive diffusion, or let's be more specific, passive membrane transport. There are basically two types of passive membrane transport. One type of passive membrane transport is just simple diffusion. Simple diffusion is when a substance passes right through the plasma membrane without any help at all. Yulia, is your question about this, or is it something else?
It's about this, but
Yeah.
just a little more specific. Could you correct me if I'm wrong? My understanding of diffusion versus osmosis is diffusion is kinetic energy making molecules go berserk and even out. Osmosis is the passing of water through a membrane.
And what do you think makes the water move through the membrane?
The difference in pressure and concentration and all that.
The kinetic energy of it. If you heat the water up, does it osmose more quickly?
Yes.
Yeah, because it's the kinetic energy of it. So you've got the right idea. Simple diffusion is basically typically the movement of something that is dissolved in water. So it is some type of solvent that is passing through, right? But if simple diffusion is 1A of passive membrane transport, then 1B of simple diffusion of passive membrane transport is osmosis. Osmosis is the movement of water down its concentration gradient using kinetic energy through the plasma membrane without any help of its own. So if you want to think of them as two completely different processes, I'm fine with that. If you want to think of them as just two sides of the same coin, I'm fine with that as well.
Okay, thank
But
you.
in both cases, simple diffusion, and if you want to think of osmosis as something separate, both of them, either the solvent or the, not the salt, the solute, not solvent. The substance that is dissolved is, isn't that right? Solute's the thing that dissolves in it. Solvent is what dissolves it. The solute, the substance that is passing the oxygen, the lipid, whatever it is passing through. Osmosis is just simply diffusion of water. But in both cases, it is passing through the plasma membrane without any help, just sneaking through those phospholipid bilayers. Okay. The other type of passive membrane transport is what we call facilitated diffusion. It is facilitated because it requires a protein. It can't get, facilitated just means helped. This is diffusion with help. Notice the help is not coming in the form of energy, because then it's not passive anymore. It is help in the form of a protein. That protein forms a hole in the plasma membrane that allows something to pass in or out. It's a door frame, right? And that's basically what a channel is. The channel would be the protein. The hole in the middle would be the pore. So whether you say channel, whether you say pore, it's basically a hole in the plasma membrane formed by a protein that allows something to get in and out. Remember, another type of facilitated diffusion is the revolving door that we talked about. Those, remember, are the carriers. Carriers are passive, but they change shape. However, to get them to change shape, we don't use energy. We don't use ATP. Instead, to change shape, they use that kinetic energy you were talking about. Oh, kinetic energy. Yeah. Of the substance to change their shape. So this was the example we talked about of a carrier for glucose, for instance, where we had glucose, and I'll draw a really simple example of this one. We had a glucose molecule that is open on one side. And when that glucose molecule binds to that glucose carrier, it changes the shape of the carrier, where it now closes at the top and opens at the bottom. And that allows the glucose to enter into the cell. Right? And remember, we gave a fancy word to these carriers. They undergo a conformational change, which again is just a fancy way of saying it changes shape. Someone raised their hand. Who raised their hand? I missed it.
I did.
Yeah.
I was gonna ask, so for ions specifically, like for example, for sodium ion, do they move either through facilitated diffusion down their concentration gradient?
Yes.
And active against? Do they use both of them?
So yes, so you've got the right idea. We're still talking passive. When we talk passive, things move down the concentration gradient. So yes, in this case, if this was indeed a sodium channel, then yes, absolutely sodium would go into the cell. Conversely, if this was a potassium channel, potassium would go out of the cell. It's passive. To get something to go against its concentration gradient, we have to use energy. And if we're using energy, it's not passive anymore, it's active. All right. Yeah, well, again, facilitate means to help, right? So if you're facilitating something, you're helping. And so this is using the proteins to help these molecules get inside, because they can't get through the plasma membrane by themselves, either because they're too big or they're polarized or for some other reason they can't get in by themselves. So they need a protein to help them. And that is facilitated diffusion. All right. Questions on that? All right, what do you guys got next? Brain's full.
Kind of along the same line of passive transport when we're talking about active transport. And secondary active transport. Can you go over antiporters and symporters, and are we also responsible for uniporters?
Yes.
Okay.
Although really, well, yes and no. So again, I like your question. So let's do that. We just did passive membrane transport. Let's do active membrane transport. And as you mentioned, it comes in two flavors, primary and secondary, because how do we define active membrane transport?
That's either directly or indirectly using ATP.
Excellent. We're either going to directly or indirectly use ATP. And if we're having to use energy to move it, most of the time, which way are we trying to move it?
Against the concentration gradient.
Excellent. Excellent. So most of the time, we are moving it against the concentration gradient. Now, to do this, always requires a protein. So all active membrane transport is facilitated, right? Because there's no way we can get something to move against its concentration gradient without a protein. Yes, Yulia.
So the previously covered passive transports, none of them are against the concentration gradient, or they can also be against the concentration?
They can never be against the concentration gradient. Remember, a concentration gradient is a hill. If you stand at the top of a hill with a ball and you let that ball go, it is going to roll down the hill and you don't have to use any energy. Is there any way you can stand at the bottom of a hill with a ball and get that ball up the hill without using energy? No, there's no way that you can do it. So the only way we can get to go against the concentration gradient is to use ATP. All right, now, as you pointed out, we have primary active transport. And with primary active transport, this directly uses ATP. So basically what happens with primary active transport is that the protein splits the ATP. And when it splits the ATP, remember, it rips off the phosphate. and that releases a lot of energy. And it uses the energy to change shape and move the substance. So we have lots of examples of these we're going to run into in the class. Some of the more common ones, for instance, there's going to be all sorts of sodium pumps we will talk about, where again, we're going to use that ATP, and an ATP I'll just use as the little yellow. But when we're splitting that ATP, ripping off that phosphate to move sodium out of the cell, or to move calcium out of the cell, something along those lines. These types, and you mentioned the term uniporters. You could argue these are uniporters, but the other common term that these are used for, besides primary active transporters, is pumps. Either pumps or they're referred to as ATP aces. Remember, an ACE is an enzyme that splits something. This splits the ATP and uses the energy. So these are typically more referred to as pumps or ATP aces for something like that.
Okay, that's where I got confused.
Yeah. The key to a secondary active transporter is that it indirectly uses ATP. Instead, what a secondary active transporter does is it uses a molecule that has a strong driving force. Remember, one of the things we talked about how sodium, because the inside of the cell is positive, sodium really wants to get in down its chemical force. There's more sodium outside of the cell, so its chemical force, it really wants to come in. Its electrical force, it really wants to come in. So the most common substance used in these secondary transporters is sodium. Sodium really, really, really wants to come into the cell. So what it does is we let that sodium into the cell, and then the energy from the sodium is used to move another substance. That's why these secondary active transporters are also referred to as co-transporters. So we have some protein. Oops, let's not go yellow. That lets that sodium that really wants to come in, come in. All right, sodium, you really, really, really want to come in? I will let you come in. Oops. But I can use the energy of that sodium coming in to do work for us. One thing I can do is I can use the energy of it to bring something else in. Like say, for instance, a glucose or an amino acid. Those are the kind of things that I would want inside the cell, so I can use the energy of the sodium to get it to do work for me. Or I can get that sodium to come in, and when it comes in, I can use it to kick out something that I don't want in the cell. I already told you I don't want calcium in the cell, or maybe I want hydrogen ions out of the cell or something like that. So I can use the energy to kick them out. So to answer your original question, when, oops, no, I want that brown, and then over here, when sodium and what it brings comes in, when they both go in the same direction, how do we refer to that type of co-transporter?
To bring some, that's a symbol.
Yeah, when they both go in the same direction, exactly. This is the symporter. Whereas when the substance moves, goes the opposite direction of sodium, which is that?
The antiporter.
Antiporter. Excellent. Now notice nowhere on any of this have I drawn an ATP, right? And that's why this is secondary. Notice these code transporters do not use ATP. Remember, what they're doing is using that driving force of sodium. Sodium really wants to come inside, and so I can get the sodium to do work for me. But what that means is for these to continue to work, as soon as that sodium comes in, I have to kick that sodium back out. If I don't kick that sodium back out, it won't want to come in anymore, and I can't get it to do work. And can I just get sodium out of a cell using HOPE? No, to get that sodium out of the cell, I have to use ATP. So here, these code transporters indirectly use ATP. The ATP doesn't bind to them directly, but we do have to use ATP to get rid of the sodium to keep this system going.
So you're using primary active transport to kick it back out.
Right, I'm using ATP somewhere in the cell to get rid of that sodium, and that's why it's indirect,
Okay.
right? These molecules themselves don't use it. Notice this is different from the most common primary active transporter. Remember the most common primary active transporter we talked about, we identified it. What is the name of the most common primary active transporter we find in a cell?
The sodium potassium, what? Yeah, sodium potassium.
Potassium pump, right? Or ATPase. Its job is to do what?
Bring sodium in and take potassium out at a 3 to 2.
You want to bring sodium in?
Maybe not.
We're using ATP. Do we really need to use ATP to bring sodium into the cell?
No.
No, so if we're using ATP, because remember, this is a pump. Notice this thing's moving two things, but it directly uses the energy from that ATP. So in this case, we're using the energy from the ATP to move two things. One of those things absolutely is sodium, but which way do we want it? If we use energy to move sodium, which way are we moving it? Out. And how many? You were correct about how many. How many sodium does it move out?
3.
3. And that same energy from that same ATP also allows us to move what?
Potassium.
And which way do we move that potassium?
In.
How many?
2.
There you go. Notice one more thing. How many so every time I use an ATP with this pump, how many positive things do I kick out of the cell?
1.
Well, you're thinking net. I'm saying how many physical do we move out?
3.
3. How many positive things do we bring back?
2.
2. So you're right. The net effect of this is we are kicking 3 positive things out and only bringing 2 positive things in. So notice every turn of this wheel makes the cell a little bit more negative. And remember, our cell's resting membrane potential is about negative 70 millivolts. They've actually looked at studies where they block these sodium potassium pumps. And when they do that to the cell's membrane potential goes up almost to negative 55 millivolts. It's almost a positive 15 millivolt change. That's how much more negative these pumps are making the cell than if you stop them. So this movement of sodium out and potassium in not only helps to make sure there's lots of sodium outside, lots of potassium inside, but it also helps to keep the cell negative on the inside, which we need for it to do its work. Remember, when a cell is at rest, doesn't mean nothing is moving in and out. There are literally thousands of things moving in and out. There is just no net change. So there you go. We did passive and now we did active. All right.
I have a question kind of on the topic.
Yes.
It was from the PhysioX assignment. I think it was like the second activity. I don't remember exactly.
Okay.
But it was the glucose and the sodium chloride. And I think you added the sodium chloride to the glucose and then to see how it would transport. And it confused me because it said the sodium was not required for the glucose transport in the simulation. And I was thinking of the co-transport when I was doing the question. So was that not what it was doing?
So okay, excellent. You have the right idea, but I can go ahead and erase this.
Sorry, I had to like double check what the question was.
No, it's a great question. So in that case, what we have is a beaker with a membrane in between it.
Mhm.
And you put a high concentration of glucose on one side, and you put a low concentration of glucose on the other side. All right. And when you go from a high to a low concentration, do we really have to use, as we talked, we've got carriers, other things like this, or really what you were doing on that experiment, either that one or the one before, is you have different size pores on your membrane, right? There are different membranes with different molecular weight cutoffs. As long as you have a size that's big enough for glucose to pass through, glucose is going to be able to travel down its concentration gradient, and it isn't reliant on that. What we were just looking at was active transport. Active transport is when we can actively move that glucose in by using sodium. That is one way we can move glucose in, and it is one of the ways we pull it out of the digestive system, but we can also get it passively as well.
And so
that one was focusing on passive transport at that point. So yes.
Okay.
Using co-transporters is not the only way we can get glucose in. Remember, we also saw the carrier, for instance, was a way that we can get glucose into the cell. So in that case, we weren't focusing on the active transport for that second activity.
So I must have just confused them in my head.
Yep.
So this isn't fully related, but does every single like cell in our body need to get oxygen and remove waste all through active and passive transport that happens within the cell membrane?
Yes, essentially. Again, if we don't have oxygen, if we don't have glucose, then we can't efficiently make ATP. We need that oxygen, we need glucose to efficiently make ATP for all of the functions. And again, we don't think of it, well, what if it's just sitting there not doing anything? But remember, as we just talked about, there are thousands of things coming into the cell. One of the things that's happening when the cell is at rest is there is a massive amount of sodium that is sneaking its way into the cell. So that means we have to constantly be using ATP to pump that out. Like something close to 25% of our ATP is used just to kick sodium out when a cell is at rest. So there is a massive, and as long as the amount of sodium coming in and the amount of sodium coming out are the same, then the cell stays at its resting membrane potential. But to kick it back out requires energy. So yeah, it's constantly using massive amounts of energy to do a massive amount of work. So every cell, every living functional cell in the body is going to require energy, oxygen and glucose so that it can be producing ATP, so it can do all the work that it needs to do. All right.
Okay, also from the PhysioX, one of the activities, I just want to double check. So one of them was to add more of the pressure. It was to, I think, activity 4, and see what would happen. And was I thinking along the right lines when I thought the pressure would push it through more quickly? And it actually did finish quicker than the rest of them, but I just wasn't sure if that was because of the pressure or if it had something to do with something else.
Right. Remember, one of the things we talked about was how things like hydrostatic pressure can increase the rate of diffusion. Absolutely.
Okay, cool.
From my memory, this is something grade-wise. I remember the labsters needed to be 80% correct, 100% complete. Was that the same for PhysioX, or the PhysioX just needed to be finished?
No, the PhysioX is basically each activity itself is worth 3 points. So if you have, for instance, this last one was 5 activities, so you will get 15 points for completing them. But I am looking for completeness. So I'm looking for that you've done all the activities, you've answered the review questions, you've taken the quizzes. I don't care if you get a question wrong on the pre or post quiz. You're not going to lose points for getting things wrong on it. But I am looking that you put the time and effort into doing it. Yes.
Okay.
All right. Any more questions? I'll remind you, as if you have questions between now and Thursday, I do have office hours tomorrow from 12:30 to 1:30 after my 431 class. Also remember, I have office hours the hour before our class. I got visited by 3 students today in my office hours, and all of them were 431 students. So my office hours, remember, are in my my office Zoom the hour before this class. So from 11 to 12, I am there. So if you have questions, you can see me at either of my office hours or email me. As many of you have figured out already, I'm very good at responding to emails. So if you have questions, email me and I will email you back. So if there aren't any more questions, last call for questions, going once, going twice.
I think I have another one. I'm just trying to
Perfect.
find.
Excellent.
Okay, it was on the Unit 1 review for
Okay.
the lab manual, and it was the one where you're asking about, or not you, but they're asking about the wounds and how to describe them.
Mhm.
And so I was getting a little bit confused on describing. I understand superior, posterior, anterior, and the difference between those. It was just, which one do you describe first? For example, like the one under his eye, like, do you say it's superior, or do you say it's the anterior, buccal, and then superior? Because I was just super confused.
Well, so here's what I would say. As long as you are using the right vocabulary, there can be more than one way to describe a wound. I mean, if from nothing else, if we have you know, a parallel wound here on my forearm, whether I go from the wrist towards the elbow or whether I describe it from the elbow to the wrist isn't significant. What I'm looking for are the regional and directional terms in response to that. So if you know that it starts in the antebrachial region, right, or in this case the cranial region, it's on, you know, I have a wound on the antebrachial region on the posterior side, right? Although, because for that you would want to say posterior side. That starts near the acromial region and goes distally towards the carpal region or something like that. Again, there isn't just one way to describe it. I'm just looking to make sure that you're using the correct directional terms, the correct regional terms when you're describing it that way.
Okay, and do you have a certain amount of the terms you want us to use? For example, I went a little bit nuts, and I used like 4 trying to like describe where it was because I was so confused.
That's fine.
So, like, can we just...
Again, I see these unit reviews as directed studying. So if this assignment encouraged you to really focus on your regional terms, then clearly you are putting the time and effort into it that I am looking for on that. Now, I also want it to be a learning experience, which is also why I post the keys so that you can see the key but again, you can see the key and see what you got right and what you got wrong. But remember also that key is going to be one way of describing it. As long as you have the regional and directional terms, if you have more terms that you're using to relate something, then yeah, absolutely. Like if you're using the mental as well as the buccal, as well as the nasal and frontal and all of that, then spectacular. Yeah.
Okay, but we don't have to. We can just use like the 2.
Again, it would depend on the question, but yeah, I'm of the opinion more is always better.
Okay. Okay.
Yeah, you could easily lose points on an exam for not being descriptive enough.
Yeah.
But you're never going to lose a point that you were too descriptive, right? So that's not going to happen. Now, if you're just throwing random things down that aren't correct, then that's not the same thing as being having a lot of detail. If you're just throwing everything you can at the wall, hoping that some of it sticks, that's different than being overly descriptive. I always say being overly descriptive is way better.
Okay.
I don't release the daily quizzes. If you want to come to my office hours, I'm happy to go over them with you. So I'll go over them with you in your office hours so that you can see them, but I do not release those questions. No. All right. Any other questions? All right, excellent. Nope. Excellent. All right. You guys have an excellent day. I have office hours tomorrow. I have office hours on Thursday. Remember, no lecture on Thursday. So on Thursday at noon, log on and start the exams. Remember, if you're being asked for an access code, that means that your Proctorio wasn't set up properly or it's disconnected or whatever. You have to set it up again. Remember to click that secure exam proctor, and then that should go through it that way. I will be available by email. So if you have any questions logging on or something like that, let me know. Remember, if you get kicked off, you should be able to immediately get back on. If you have any problems with that, contact Proctorio's tech support. I cannot put you back into the exam. So the first person you want to contact is not me. Contact them. All right. You know what to expect. You've seen a lab exam now. You've answered multiple choice questions before. You've answered essay questions before. You guys know what to expect from this. I have faith in you. I know you're going to do well. But the one last thing I will remind you about is the key to this class. One of the huge advantages of this class is that with 5 lab exams, 5 lecture exams, and a final exam, no one grade is going to make or break your grade in this class. I can tell you now with 100% certainty, no matter how you do on the first lab and lecture exam, mathematically, it is still going to be possible for every single person in this class to get an A. In fact, remember, your final replaces your lowest lecture score. So even if you got a 0 on your lecture score, which you would have to go out of your way to do, you have to physically avoid this material and purposely not do well to get a 0 on your lecture exam. But even if you did get a 0 on your lecture exam, you could still get an A in this class because you could replace it with the final. Use this as a learning experience. Be smart, put your effort, put your time into it, be descriptive, but also know that this isn't life or death. All right. You are going to use it as a learning experience. If you do well, awesome. You know you're on the right path. Continue to do that. If you don't do well, know you have plenty of time to learn, change it around, and still get the grade that you want. All right. And by plenty of time, I mean 2 weeks till the second exam. All right. Excellent. Good luck on Thursday, and I will see you next Tuesday in class. Take care, guys.
Bye.